We're going to start the afternoon session off with the topic, small as it might be, approaches to future clinical trial design, and I'm going to turn it over to Prem Soman, who will be moderating, and there are a few presenters.
Thanks again, Matt. So this is a big session. We have a number of speakers and a number of discussants. So I'll try my best to keep to point and to time. So the first speaker of the afternoon will be Adrian Hernandez from Duke University. Adrian.
All right. So thanks, everyone. So hopefully I can wake everyone up after lunch. So I'm here to kind of set the stage. And so I think what we took home this morning is that there's been a lot of advances around amyloidosis, but still so many challenges. I think the patient stories really told it, is that while everyone is optimistic about the future, there are people who are still suffering with amyloidosis, and so now's the time in terms of really seizing the day. There are, however, distinct challenges. So the concepts of having placebo-controlled trials is an issue. It's a major concern. Access to therapies is also a major concern. Timely access for evidence generation is a major concern. And so I think in this next part, we're going to hopefully tee up some of these issues and get different perspectives added in here in terms of different approaches. So these are my disclosures, and I have worked in the past with companies that work in the space, as well as the Duke Clinical Research Institute. So I do think it's a simple question of alignment, as that when we think about endpoints, and this is what I'm kind of teeing up, like the end in mind, what the outcomes that we're really trying to assess and achieve. And so if you think about an endpoint, really trying to see what are the key characteristics does it fulfill? Is it measurable? Is it meaningful to patients and clinicians? Is it reproducible and valid? And there are a range of domains to consider: clinical events, patient symptoms, function, or surrogates. And importantly, it has to be regulatory enabling to establish substantial evidence of effectiveness. There's also one thing that's not on the slide, and is that it also has to be enabling for payers. And so there's going to be some considerations here that while we may be focusing on the regulatory side, that may not necessarily be the last mile, as we do need to ensure that the payment systems around the world are convinced by the evidence that we generate. We do, fortunately, have a lot of different endpoints in our toolbox for outcome assessments. And so here later you'll hear a little more in depth about some of these areas, but I'd like to categorize it in these domains, and also think about what can be expanded on in terms of recent developments. So there's patient-reported outcome measures, clinician-reported outcome measures. You can have neutral observers, and we actually saw that in some of the testimony with a patient and a spouse, noting how that person was doing. You can have performance or objective measures in terms of how people are doing, and then also you can imagine a variety of ways of taking advantage of digitized data to understand someone's health. And then as we consider patient-centered outcomes, not necessarily just patient-reported outcomes, but I'm just invoking patient-centered outcomes, things that are meaningful. And I put this in a set of generalizable categories, and we heard that and saw that from some of the data, that there can be different aspects to someone's health that has a range of symptoms, and how you combine this is really important. And some of these categories have direct relevance to the story of amyloid. Function is really critical, and we heard a variety of areas that people care about in terms of how they function, and that is important to integrate as well. And all this can be combined in terms of different assessments of domains of quality of life. That includes mental health, social aspects of health, well-being, and satisfaction, and different elements here. And so it's this holistic picture that I hope we also consider. So there is a need to say something about surrogate endpoints, and part of the charge was even asking, like, hey, could increased diuretics be a surrogate because it's predictive of worse outcomes and may be clinically relevant because sometimes that's what we use as a simple surrogate to judge whether someone's getting worse. And we're fortunate that the FDA does have a history and guidance about surrogate endpoints. And I just put the definitions here so we can just remind ourselves. It's a marker. It can be a laboratory measure, radiographic image, sign, or other measure. That's not a direct measurement of clinical benefit, but is known to predict clinical benefit and can be used to support approval, or is reasonably likely to benefit. And you can look up at this table surrogate endpoints that have been used in the past for drug approval and licensure. And so there's a long history of this, and so often it has to deal with issues around rare diseases, whereas there's a lot of complexities in terms of getting to other outcomes that people are most interested in, but you need to be able to make a decision on the best evidence, and so this can be a pathway to do that. I'll have to note my personal bias is that I usually don't like using drugs as a surrogate endpoint because that can be influenced in all sorts of ways. So I think there's other ways to consider this in this space. And then I think there's a lot of interest in terms of combining outcomes in terms of having a picture of the total journey of health or composite outcomes. And so stealing a bit from my colleague, Chris Lenzel, who's heads up data science and biostatistics at DCRI. And so in the hallway, we often have debates about what's the most meaningful to patients. And so he comes at it as a data scientist saying every data point counts, and I come at it as a clinician saying, well, perhaps, but there are some that count more. And so how do we do that? And so there are ways to consider this in terms of composites of hospitalization and death, days alive and well, progression to the next level of care, or regression to level of care or health and well-being, and then various methods in terms of rankings or comparisons for different components. And note that, in general, one has to assess the benefits to risk, and so safety is a separate category. But this can be a relatively a common approach to have a composite, and there are some pros and cons to it, such as you may not be able to discern what's truly happening if you had a composite of, say, 20 outcomes as opposed to 3. Or are there other ways that we can do better? And so in considering this in this case, really thinking about what are the key attributes here, so that we should have something that obviously gives credit for a treatment that has good outcomes that matters to patients, goes down or is penalized when that treatment causes a worse outcome, has the way to so-called untie things, meaning that every participant counts and there are few ties in the schema, so that if you compare it to some of our traditional trials where we make a decision on 80 or 90% of the patients, that can you reverse it the other way, where you're making a decision or an inference based on the 80 to 90% of the patients. And is it measurable over a relevant time course, meaning it doesn't take decades, that actually can be meaningful to clinicians or patients, and isn't clouded by other things that come in. And so I think in this space, that's especially important given the rapid advances that we're seeing in the field. So one other category is just, does time matter? And so I'll say that the answer is yes, but I'll be interested in what you all think. And so if you look at, say, a patient experience, every day, every week, every month, every year matters. And so while we, like me and some of our clinical trials, think about a death date as an outcome, there's a lot of things that lead into that. And so we're seeing more and more interest in terms of assessing outcomes over time. And, this is a generic illustration of this, but you can imagine a scale that incorporates kind of severity of symptoms, but also includes a major competing risk, such as death and hospitalization. And we put this in place in some other programs unrelated to amyloid, in part because you had to. COVID was one of those examples where it forced decision-making. And so a program called Active 6 that we were guided by FDA in terms of what would be meaningful and also timely. We had a combination of measurements that included symptoms as well as health outcomes. And because the pandemic was ever-changing, it was a way to also assess if there was something meaningful as needed. So in summary here, I hope we keep it real, meaning keep endpoints that are measurable, meaningful to patients, clinicians, et cetera, and consider how you can make every day count. I think that may be really important. The stories that we heard this morning, to me, just underscore that. And so I think there are ways to do that with these different domains. I do urge a word of caution about surrogates and that we've been burned before, but I think the field here is evolving such where we're seeing greater understanding in terms of biomarkers and imaging. There's a range of possibilities here. So I'll turn it over to the moderator, next speaker.
Thank you, Adrian. Thanks, Adrian. So next, we'll hear remotely from Pablo, Dr. Garcia-Pavia from the Spanish National Cardiovascular Research Institute.
Hello, everybody. Thank you very much. I'm going to share my screen. First of all, I would like to thank ARC for the invitation to participate, and also I'm sorry not to be there. Okay, so I was told today to talk about future approaches to clinical trial design and discuss specifically about survival. Okay, so these are my disclosures. Well, we have seen that the prognosis of these patients has dramatically changed over the last 20 years. And here there is a graph from the National Amyloid Center showing how the median survival has improved over time. And certainly this is because we are diagnosing patients earlier, but also the effect of including patients in clinical trials and the therapies that we have available. Of note, tafamidis, which is the main drug used for TTR cardiomyopathy patients, is not approved in the UK, so very little number of patients included in this graph were treated with tafamidis. Okay, and certainly this we are seeing those data coming from the National Amyloid Center in the UK. We already knew that something was changing, and we had data from the placebo-treated patients included in the track, but also natural history studies, again, from London and the Mayo Clinic, showing that the survival that we were witnessing in our patients was better than the ones that we observe in the placebo-treated patients of the track that, as you might remember, as it has been said today, were recruited between 2014 and 2015. And probably this is the explanation from some of the surrogate endpoints that have been used in some of the trials, like the attribute cardiomyopathy trial, where it was found that the change in 6-minute walking test distance at 12 months was unaltered between patients who received the drug and patients who were treated with placebo, and this was remarkably better than what was found in the placebo-treated patients included in the track, which certainly we believe is because of the type of patients that were included in the trial. And certainly there were around 5 to 6 years distance between both trials. Interestingly, if we take out the effect of the modifying therapies and the clinical trials, and this again are data from the National Amyloid Center, we can still see that although the curves are a little bit worse, there is a remarkable difference between eras. And everything can be even can change if these patients had been treated with tafamidis. So we certainly have a big problem if we want to focus in survival in these studies. And what are the alternatives in this changing world? Well, we can have larger trials with higher number of patients. And certainly we are moving towards that direction. If we see the number of patients who were enrolled in the TTR transform trial in comparison with the ones who were with the number who were enrolled in the track study. We can have larger trials, and that's a possibility, but obviously that has also problems because it's gonna make trials more expensive, and certainly what is more important is gonna delay the availability of some new treatments. But that's certainly something that is being done. And again, comparing what was the duration of the track study and what is the duration of the TTR transform trial, this is another possibility. And then we can include sicker patients. I'm going to comment on this a little bit, okay? So we can focus in patients who are sicker and see if what is the effect on these patients of the new drugs. But how do we do that? Because if we do that, we do have staging systems, the Mayo Clinic staging system, the Columbia staging system, or the National Amyloid Center staging system. But all these systems were derived in a time when we did not have baseline therapies or the therapies that we are using currently. So do they capture fully what is the prognosis of our patients? Are they good ways of addressing the prognosis of our patients nowadays? Certainly, there is a limitation of the possible number of patients available in a competing world with so many therapies going around, so many trials going around. So including a subset of these individuals might be problematic, because if we focus only in the sickest ones, there might not be enough sick patients for upcoming trials. Moreover, this increases the risk of negative outcomes in the trials, because if we focus in the sicker ones, it's always a risk in the sense that if they are too sick, they may not get benefit from the drugs that we are developing. And also, and this certainly worries me a lot, is how do the regulators will see these trials at the end. Even if they show benefit, there might be some possible limitations regarding authorization or maybe afterwards reimbursement in the less sicker, because those ones will have not been included in the trials. So can we propose that if these drugs demonstrate usefulness in the sicker ones, can we assume that they're going to be useful in the less sick patients? That's something that maybe we can discuss afterwards. Or we can decide that mortality is no longer a possible primary outcome. And that might be the way to move forward. We can try, as it has been discussed, to incorporate composite endpoints. And certainly we have the example of heart failure trials in this sense, and quite big trials that have been approved in some areas where there were no therapies available, like HFpEF. And here we have the example of the DELIVER trial with dapagliflozin in this type of patients, a massive trial with over 6,000 individuals, where the cardiovascular death, which was a component of the primary composite outcome, was not significant, but regardless of this, the therapy was approved. And we can see that even overall mortality was not different between groups. But because the primary outcome was statistically significant, as you can see here, and maybe because HFpEF did not have any available therapy which had been shown to be useful, maybe this was the reason why this drug, one of the reasons why this drug was approved. But even in another completely different scenario, which is HFrEF, where we already had several medications that had shown mortality benefits, we had the example of Vericiguat, and in another quite big trial, the Victoria trial, with over 5,000 patients enrolled, where we had similar findings: death from cardiovascular causes in the primary composite endpoint, or taking into account all the deaths, cardiovascular deaths, in the secondary outcomes, did not result significantly beneficial to the patients treated with the drug. And regardless this, because the primary endpoint was in favor of patients treated with Vericiguat, this drug was approved by regulators for the treatment of patients with HFrEF. So certainly this might be the way to move forward in this disease in the coming years. And that's it. Thank you very much.
Thank you, Pablo. The next speaker will be James Signorovich from the Analysis Group, Inc.
Thank you. So while it's coming up, I'll just introduce myself. My name is James Signorovich. I'm chairing the statistical working group for the forum and really grateful for the opportunity to talk to everyone today. So I'll be speaking about functional and patient-reported outcomes and what we might need to incorporate those into trials of ATTR amyloidosis therapeutics. I think the rationale for why we'd want to consider a host of other functional and patient-reported outcomes is pretty clear. We heard, I think very clearly from the patients, just the multiple organ systems, sets of symptoms, and how those impact life for patients with this disease. And so, we ideally want to be able to measure the whole impact of the disease and the full benefit of treatments. I think we'd all agree with that. The concern, of course, is how do we do this without making life harder for ourselves, especially from a clinical regulatory perspective. So there's a number of measures that have been informative already in trials in amyloidosis. And as has been discussed, they span the range of performance-based outcomes, patient-reported outcomes, clinician-assessed outcomes. Some limitations, though, are we don't yet have a validated single metric that addresses all domains of the disease, or kind of the type of data that we would need to know how to incorporate such a metric into a trial, considering that patients are going to come in heterogeneous at baseline in terms of which domains are affected and the extent of impact and the degree of potential improvement that could even be possible. We're also importing some measures from other therapeutic areas that have a long track record of data, but we may not understand how well, exactly what they mean in the context of ATTR amyloidosis. And then finally, as has been emphasized, natural history is changing. So I'll just zoom in for a bit on 6-minute walk to kind of illustrate some of the challenges that come up as we try to use these metrics in trials. So we, in general, we think we know a lot about 6-minute walk just from all the different cardiac and neurological and neuromuscular indications that it's been used in. But ATTR amyloidosis is just very different from these other indications. You can see on the right-hand side just some baseline ages and 6-minute walk distances from 3 phase 3 trials in ATTR-CM. Patients are coming into these trials with older age and much worse initial 6-minute walk distance than we see in a lot of heart failure trials. ATTR is, or at least used to be, more rapidly progressive, declines in function. And also the deficits that we're seeing in 6-minute walk are due to a combination of impacts. Some of it's cardiac, but it could also be neurologic. We heard today that amyloid can get into the lungs, so there could be pulmonary impacts. Constipation or pain could also affect walking distance. So the things about the disease that are being measured by the performance on the 6-minute walk test probably differ quite a bit from what's being measured in other therapeutic areas. And then finally, I think we've seen and discussed extensively just the changing nature of the natural history of amyloidosis, and that really shows up in 6-minute walk with effective treatments, modifying the trajectory significantly, and kind of recent increases in broader diagnosis and earlier diagnosis, kind of changing the set of patients who are going to present and enroll in clinical trials. And so I think it's helpful to kind of zoom out and think, what does this mean for someone who's sitting down to design a trial and hoping to incorporate, 6-minute walk or KCCQ or MNIS or all of the above as a clinical outcome. So for those of you who've sat down to design trials, and this was alluded to a bit earlier today, there's just a whole bunch of decisions that need to be made to specify who's going to get into the trial, what we're going to measure, how long we measure it for. And there's some luck involved. I heard luck invoked a little bit for ATTRACT, and luck is always great, but ideally we'd like to inform these decisions as much as we can with evidence. So if we're thinking about 6-minute walk as an endpoint, what should our inclusion-exclusion criteria be in light of recent trials that have used 6-minute walk as an endpoint? Should we have inclusion-exclusion criteria that try to enrich for patients who are going to have a particular rate of expected decline on 6-minute walk? Prognostic factors are critical. We've seen there's a lot of heterogeneity in rates of progression. If we can explain that heterogeneity with baseline prognostic factors, that really gives us a key to design better clinical trials that can increase power, have smaller sample sizes, and either enrich for or stratify on those key prognostic factors. How long should we run the trial? Is 1 year enough? That could depend, again, on the population and the expected rate of change. And then once the trial reads out, and even when we're designing it, we're going to have a sense of what is a meaningful change for the 6-minute walk distance in this population. Now, again, we have a lot of data on that from other therapeutic areas, but how well is that going to translate to ATTR amyloidosis? I was really struck hearing the statements from patients earlier about how they valued kind of particular increments of function that allowed them to do things with their family that were truly important to them, while accepting that some other things that they would have liked to have done are off the table. So I think these kind of the value of those incremental preservation of function just came across very profoundly, and that's something that if we want to understand what 6-minute walk means in ATTR, we're going to have to get the data to really connect that back to the value to patients. So as we're sitting down to design a trial and we have all these decisions, we really have to think about what kind of evidence do we need. Like, any of these decisions could end up being make or break for the trial, and we want as much evidence as we can. And so kind of listing out the things that feel like they're urgently needed, I think one is just basic descriptions of natural history. What does change look like for 6-minute walk, MNIS, KCCQ, and how variable is it? And how do these measures relate to each other? Just basic descriptions that are up to date and in representative populations are sorely needed. Prognostic factors, I can't emphasize enough how much of a key these are to better clinical trial designs in diseases that are heterogeneous and progressive. There's really no better way to inform a design that's going to be efficient and minimize the duration and number of patients who need to be on placebo than to really understand the prognostic factors. And we know a lot about prognostic factors for hard events, for cardiovascular events and mortality, but we shouldn't accept those as like the end of the story for prognostic factors. We need to understand what predicts changes in 6-minute walk or changes in KCCQ in different populations. We talked before about clinically meaningful thresholds. There's probably space for new patient-reported outcomes or scales that capture the totality of the disease. And to do all of this, we really need clinical data. And I'll just emphasize two final points. As we generate these descriptions and learn from natural history and placebo arms, it's really critical that we do this in a way that's quantitative and feeds into clinical trial designs. And that really benefits from getting the right people together to design the studies, work through how to look at the data so that we're doing things that are going to be useful for the trials in the end. The publications are critical, but a lot of times they get us maybe 50 or 80% of where we need to be for designing the best possible trial based on the data that's available. Quantitative insights are critical because at the end of the day, trials are a quantitative exercise. We also need the evidence and data to be timely and accessible. There are so many situations that are just very frustrating where, there's data sitting out there, there's trials being designed, and you kind of know that if the design of this trial could benefit from the data that's already available, it would be a better design, right? The drug may work or not work, but what we really want to happen is the trial to tell us as definitively as possible whether the drug works or not. If the trial fails and we can't tell whether the drug works or not, that's kind of a disservice to everyone. So I think being really creative about how we can, if possible, share the actual data. But if that's not possible, how can we get the right analyses done in a timely way and in the hands of the people who need them to design these studies? Thank you.
Thanks, James. And the last speaker will be L.J. Wei from Boston, from Harvard.
Thank you. Let's see. Let me share the screen. Can you hear me all right? Thank you very much. First, thank James inviting me for this prestigious conference. This reminds me a few years back, I was invited to attend a FDA advisory committee meeting talking about non-inferiority trials for antibiotics. So each company probably get 10 minutes to really state their position. So the sponsor, I was invited to help this client ask me to give a 3 minutes talk. So I asked the guy, say, usually I give a 30 minutes talk. How come I got only 3 minutes? The guy say, L.J. Wei, you don't understand. If you got a 1 minute time with FDA or EMA, and that equivalent to $1 million. So you already got a $3 million. So today I got a $6 million. So probably I can cash in and retire. Now, I think everything we're talking about here is very interesting. It's involving a terminology we call estimand, E-S-T-A-M-A-N-D, which probably most of you are familiar with this terminology, which is a collective word, including how to define the target population, how to define the endpoint, how to define the treatment difference, how to handle intercurrent event. So let me give you one example today. It's a long trip to come here, actually, long way to come here. Pablo talking about composite endpoint, Andrew also talking about composite endpoint, which actually came a long way. So I give you one example, illustrate how difficult the clinical trial methodologists come here today. Now, the conventional wisdom, as we heard, you have efficacy and safety, right? First, for example, efficacy, what is the usual way to do it? You define a primary endpoint. Then you define the several secondary endpoint. Then you can define other endpoint if you want. In some study, you probably have 20 endpoints towards the end. And then safety, you're going to summarize the AE, SAE in different categories. For example, CV related or GI related, and etc. Now, we designed a trial using this primary endpoint, for example, survival. And you say, all right, I wanted to hazard ratio 0.5, so I need 100 events. So I'm going to get on the trial, figure out how many patients. End of the day, unfortunately, in this setup, the p-value plays so important role. I don't know why, but we still, if your p-value less than 0.05, you say, wow, I got it. But if your p-value is a 0.07, you say, wow, sorry, maybe you'll have to thinking about next trial. Maybe you see your next life. And if your primary endpoint didn't make it, the secondary endpoint doesn't help at all. So how to analyze those data? We analyze data for each endpoint. We do the separately analysis. So you have efficacy, each efficacy endpoint, you give me a summary statistic, like hazard ratio, whatever it is. Then you have safety, you give me another summary. Then you use those two numbers or several numbers to decide the risk-benefit assessment, which to me is so difficult. Why? Because we lost this correlation in the patient level. At the patient level, for example, safety and efficacy, right? We don't really know. Some patient benefit a lot, maybe has to suffer a little bit of toxicity, which probably is all right. But some patient probably don't get any benefit, but got a pretty bad safety issue. But we totally lost for that information. Now, if you look at this clinical practice, if I were a patient sitting in your office, you look at me, say, in the past 3 months, I give you this particular drug, how do you feel? Well, the doctor must check my blood test and they say, how do you feel? Anything, bothers you? I say, well, gee, I cough every night, I cannot sleep. Then the doctor said, well, maybe that's not good. Even though your blood pressure is under control, I probably have to change the treatment for you, right? You notice this interesting phenomenon means the doctor in practice utilize the patient level, the multiple outcomes, make a decision, right? But on the other hand, in clinical trials, somehow, we don't do that. For each outcome, we do separately. And we get several summary statistics that you say, well, why don't you tell me, do you think it is a benefit over the risk or the other way around, right? So here is one thing, if we can actually come up with a composite, like Andrei Pavlov talking about, in the patient level, and can we talk about the so-called net clinical benefit, right? For example, safety and efficacy. So we're talking about totality of evidence of the treatment effect. Now, let me share this with you. There's a deliberate trial Pablo just talking about. In fact, we just published using that data. I can just talk about later. But this Paragon data is interesting. Paragon was a trial, probably most of you know very well, randomized, compare a combination treatment with a Vasartan. The question is, do you think this combination drug would improve the patients, right, preventing from hospitalization, heart failure related, or CV deaths? But ejection fraction is a 45 or above %. The primary endpoint is including recurrent event, hospitalization, not only the first one. Okay, so here is a typical patient profile. The patient number 1, you notice patient have a red cross, that means hospitalization. The circle means the patient censored. That means towards 48 months, we actually study terminated. Second patient did not have any event, but a censored. This guy entered the trial a little late, for example. Third patient had hospitalization, then death, based from the CV death. That's a black dot. Fourth patient didn't have hospitalization, but died from a non-CV death. Okay, this is a typical heart failure trial, and we have hospitalization and the death, CV related or CV related. Now the question is, can we actually utilize each patient's the history, 48 months, come up with a interesting interpretable endpoint? Can we use that endpoint to actually evaluate treatment difference? So here's one approach. Now remember the case number one, the patient number one, this patient had 3 hospitalization, then censored, right? So I'm going to plotting a cumulative number of events over time. Horizontal line is the number of months follow-up. The vertical, the scale is 1, 2, 3, first event, second event, third event, and etc. So every time I have experienced event, I'm jumping up 1 step. So this patient has 3 jumps, then censored. Then I ask myself, say, well, gee, you know, this is interesting. Can you actually utilize this curve and give me a summary endpoint reflecting how good or the progression of the disease or the disease burden over time? I say, well, yes, the higher curve, the worse. So if I use area under the curve and sort of summarize how good you are, I say for this patient, area under the curve is 72 months. I say, well, gee, that's interesting. I know statistically speaking, you tell me area under the curve is a good summary, but clinically, how to interpret this area under the curve? I said, well, you know, this is very interesting. The area under the curve means the cumulative event-free time lost, right, during the 48 months follow-up. So for example, the first patient, so 12 months, 48 months, 36 months lost, right? So this patient from the first hospitalization, the event-free time lost 36 months. The second one is 24 months. Third one is 12 months. You add it up, 72 months. So the case number 2, this patient censored. So we don't have any jump. And the third patient, the patient died of a CV event, CV death. So you have 2 jumps. Of course, you can argue, I should have jumped in a big size if it's a death, right? Not hospitalization. Then we can talk about how to actually penalize even deaths compared with hospitalization. In any event, you notice this each patient, I got a single interpretable, clinically meaningful summary. Then I said, could you collectively and in group-wise, how about treatment A, treatment B, and etc. Okay, I said, forget about the censoring. We're going to summarize this one using the so-called cumulative curve. Now, how did you read this curve? The horizontal line, again, is 48 months. And the vertical scale is a cumulative numbers, right? The mean numbers. So the blue curve is a Vasartan, the monotherapy. The red curve is a combination. You notice the higher the curve, the worse, right? Because you have more events. And the area under the curve is averaging of the time lost. So in the Vasartan, on average, patient lost 1.10 years totally, cumulatively, but the combinations lost 0.95 years. Okay, so I say, wow, that's great. Can we actually do some analysis? The answer is yes. So this actually, the methodology I presented here was published last year in the New England Journal of Medicine, a new journal called Evidence. And you'll probably notice some of your clinical friends is on the co-author list. So before we give you the result, how to compare AUC between the 2 arms, how actually people dealing with those kind of recurrent events analysis. Now we have so-called Anderson-Gills, that's older paper, long time ago, and LWYY is the paper I wrote back in 2000. In fact, this paradigm using this methodology, I'm going to share with you just very quickly, and etc. I also listing win ratio and Finkelstein and the Schoenfeld test, right? There's some issue about the win ratio we can talk about later if you'd like to. So let's go back to the paradigm. There were 2,407 patients and 2,389 patients assigned to combination and the single arm. And you have 690 and 797 heart failure hospitalization. And also 405, 433 had at least one. That means you had 2 or 3 heart failure hospitalization. And for CV deaths, those two groups are pretty even divided, 204 and 212. And non-CV deaths also pretty even. Okay. Now how the paradigms methodology they use a so-called LWYY. What is the LWYY did for us? They're assuming these two curves are actually the ratio of the two curves over time is a constant. It's very much like hazard ratio concept, right? And if you're assuming this constant ratio across the time, that's what paradigm did. So the, sorry, let me do this. Constant over time, the ratio is 0.88. That means the combination drug will reduce 12%, right? The so-called disease burden. And the p-value, look at the p-value 0.08. The stratified version, the p-value resulting 0.06. This is actually very interesting. New England Journal of Medicine published a paper in the conclusion section. The only one sentence, they said, well, this trial didn't make a statistical significance. That's the end of story. It's a very interesting to notice people still treating 0.05 like a magic number. But we're not so sure the constant ratio assumptions are valid. In fact, several regulatory people asking, do you know how to checking those proportional sort of hazard, right? Proportional constant assumption. We actually don't know how to do that. Of course, you can do goodness of fit test getting p-value. As you know, model checking with p-value is useless, right? So the idea is don't use any model. AUC is very interesting without any modeling. If we use a ratio of 2 AUCs, like we're talking about, the ratio is 0.86. Now remember, using the other methods of 0.88, it's very close. And this is a confidence interval. Look at this p-value. Just turns out to 0.049. Wow, we said, well, wait a minute. So my friend Scott Solomon was a PI. He said to me, he said, if we knew this methodology, we can use this yield p-value, not a 0.06. But I said, well, it's not too late to talk about it. And the difference between those two is 1.8 months, the total so-called time lost. Now, one problem about this methodology, when terminal events, in this case is CV deaths or the ending deaths, right? Now, if they have a different so-called distribution over time, then you notice, right, if a group of patients have a shorter survival time, of course, the number of hospitalization will be artificially smaller, right? So this kind of methodology doesn't work. So we need to figure out another way to analyze data. If you have a differential dropout, patient dropout between the two groups or the different survival time. Now, let me just close up. As Andrew mentioned about so-called the off-hospital survive, I think this is a very interesting idea. We ask ourselves, say, well, during the 48 months, right, can you actually count how many days the patient survived, but they are out of hospital, right? This is actually a paper written by our FDA friends. I believe they're probably in the audience. And they actually use this endpoint to analyze, to compare those two treatment in the paradigm, and also CHARM trials. Now, someone actually asked, well, even the patient off the hospital, maybe their life is not that great. Pablo talking about survival, of course, we hope everybody survive long, but we're also hoping people survive with happiness, right? Have a good life. instead of a miserable like a cancer patient that lie on the bed every day. So we're talking about almost like a disease-free survival, this concept. So in the cancer, we're talking about cure concept. So we have to use this quality of life, functional measurement, whatever the patient outcome, reporting outcome, and to actually mingle with those so-called composite endpoint. Of a hospital, we can easily using a Apple Watch, for example, right? To reporting every day to say, hey, do you have a good day or bad day or okay day? And use a very simple device, catching up an essential quality of life. And we use that one to say, hey, how many happy days when the patient survive? So I think this is just giving you a sort of a feel how long. This actually, Paragon was designed 15 years ago. And I think in 2019, finally, they got approval from FDA. So you can see it's a long journey from your design stage to the analysis stage and also end the product. So let me stop here. Thank you so much.
So thank you for that really thought-provoking talk. The plan was to dive straight into outcome measures and a discussion around those. But this morning, we heard some really moving patient stories. And one of our speakers, Michael Lane, made a really impassioned plea for rapid approval of drugs in this, rare disease but rapidly progressive disease space. And so we have a challenge. All of this morning, we talked about the changing landscape of the disease, earlier diagnosis, and the need for more patients and longer studies to come up with some meaningful hard outcome differences. And on the other hand, we have this plea from our patients that, we want drugs approved more quickly. And so how do we reconcile these two needs, this challenge? And so I thought if this is okay, we have Norman Stockbridge, who's the director of our drug development in cardiology and nephrology. So if it's okay, sir, I'm going to direct that question to you.
Yeah, great issue. I think there are actually three competing interests. There's trying to get the answer more quickly. There's trying to get the answer with spending the least amount of money. Some people care about that. And then there's having assurance that you've actually got the right answer, right? So all of these things sort of compete with one another. I think we have to deal with those competing interests. I think we try to keep some kind of standard with regard to the evidence supporting a claim. And I think, I think it's a reasonable community discussion about how those things play off against one another. Are you going to be happy if the next generation of patients are taking therapy that isn't well established to have a clinical benefit? So other people should comment on how they view the balance among these things.
Are there sort of existing pathways for expedited regulatory approval in uncommon diseases and very rapidly fatal diseases?
So in the United States, there's only there's full approval, regular approval, and there's accelerated approval. Accelerated approval is based on a putative surrogate, one that's not well established to predict a clinical outcome, and so it carries with it a requirement to validate that outcome in the post-marketing setting. That's harder than it sounds. You can look around at things like, maybe, levels of circulating amyloid protein. And, it has a certain plausibility to it. How does it relate to outcomes? It's how you think the disease manifests. But in order to confirm the clinical benefit at the end of the day, you've actually got to have a pretty well understood relationship between your biomarker and the outcome. And that's where it gets difficult, is developing the evidence base that would enable you to have a good sense that if you've got an effect on the biomarker of some magnitude, that will likely translate into a clinical outcome of some magnitude, and that informs how you have to size the trial, the outcome trial. But those are the only two mechanisms we have.
Any other comments from either people online? Clemens, you have your hand up.
Yes, it's similar in Europe. Of course, the demand of the patients and to have drugs fast approved is understandable. But something that they mean but don't say is they don't only want to have it approved. They want to have drugs approved with a positive proven benefit risk balance, and that is our task. So it's not only be fast, but be sure that benefit risk balance is positive. In Europe, we have a similar, it's not accelerated procedure, it's a conditional marketing authorization, a little bit on a lower standard, but still at the time of the conditional marketing authorization, the benefit risk, a positive benefit risk should be clear. And it should be clear that the data that is missing can be generated, and that is sometimes a problem, because if you have an approval, a conditional marketing authorization, we publish that. The main outcomes will be published, and then the patient may not want to stay in a controlled clinical trial, drop out, and then the result may be that we never will see the final results of an ongoing trial. So there are pros and cons of this conditional marketing authorization, and of being a little bit earlier on the market versus being a bit later, but having a good data on benefit risk overall in subgroups and so on. Thank you.
I may add. Oh, go ahead.
I was just going to add, I think this is always a very difficult conversation with patients, so I can understand why someone who has a life-threatening disease wants to get access to the drug, compassionate use, give it to me now, I want to get it. I have to say I always struggle, if I give it to you, why shouldn't I give it to everybody else? And if there's no one who enrolls in the trial, what I don't think patients want is just access in the short term. They want access to highly effective, proven drugs for the long term, and that's not going to happen unless we do clinical trials. So it's a hard discussion, but I think it's part of what we need to explain to people. This is the process that we have to go through to make sure that what we're developing is truly, in the long term, safe and effective for not just them, but for everybody.
James?
Yeah, I wanted to bring up the topic of enrichment and prognostic factors and rates of progression. If there are subpopulations that we can identify as faster progressing, there would be an opportunity to do a shorter trial for that population. I think there's a tension there, because if a drug is effective, we would want that to be accessible for the broadest population that could benefit. But that might confronting the fact that there's heterogeneity in risk and rates of progression might be part of the story in finding out how to accelerate the testing of drugs.
Thank you. Adrian, you had a comment?
Yeah, so I guess, norm, of course, makes it simple, makes it sound very easy, just three competing interests, and we can align and solve that. But I want to add a fourth one here, which is, actually, in this space, is some ethical dilemmas I think people are going to have to wrestle with. And so in general, people are going to seek to do placebo-controlled trials because it's the easiest way to show definitive proof. However, to do so, you'd have to go to communities, as I noted this morning, that don't have access to, say, a standard of care that may be representative in, say, New York or Boston or even Durham. And so that's an ethical dilemma, I think, is that, you can see the arbitrage for doing so. If we don't solve this, there's other ways that people, just thinking about different types of trial designs, which is, one could do in this population historical controls, and so having objective benchmarks that could be used. And someone mentioned the device world, that's been done before. There are also other drugs that have been done in that way. Non-inferiority designs can be done, but that doesn't solve the cost issue because the cost issue goes up typically for those types of studies. And then there's also a bit atypical designs, which is not reflective of clinical practice, because in this space it doesn't seem like add-on therapies is what we'd have. Like, unlike in heart failure, we're used to HFrEF or just add on, and we have no idea actually what's the right therapy. We just add on. It could be actually, instead of 4 things, it could be 1 or 2 things, but we have no idea. And so in this space, having designs where you can have, in addition to what the standard of care is, but then having randomization for withdrawal, so you actually know if there's a sustained benefit or is there an additional benefit. And so I think, for this, the constraints here, it is going to have to take some, I'll just say, creative thinking and creative approaches that may not have been traditionally done in clinical trials or in the regulatory setting.
So, Adrian, you mentioned historical placebo data as comparative. Do you think that, we spoke so much this morning about the changing landscape and the natural history. Do you think today we can use historical placebo data for TTR?
Well, I think it's harder, because if you look at just the last 5 or 10 years, and people talked about this earlier, is that we don't necessarily have yet this kind of Eureka data platform that has all the imaging, all the biomarkers in one place, and so that would be great. And so that could be helpful. The other thing is just thinking about this in a little more global fashion as say, tafamidis has greater access, and you start having, say, could have worldwide registries that are coordinated, then you can start, having, say, a much more robust concurrent control, so you can see when people are getting initially diagnosed and then getting what their initial treatments and longitudinal health is, and then when they get access to therapy, that could be one way. But certainly, having contemporary controls or historical controls of objective benchmarks could be good.
Sure. Please.
Wiley Chambers. I would just like to remind people there are a number of endpoints which you cannot use historical controls for, things that are effort dependent. And in my field, which is ophthalmology, visual acuity is a readily available endpoint, but it's effort dependent. People can do better because they try. Historical control setting, people don't try. They just give whatever they think they can go and read. But when they think they've received treatment, they push it, and you get very different results. And obviously, visual acuity is not the only endpoint that occurs in.
Yes, and then Matt.
Okay. Fran Cunningham, Department of Veteran Affairs. I run the Center for Medication Safety, but also responsible for looking at outcomes assessment after drugs have been approved. And I wanted to bring up something that was mentioned earlier, and that's real-world data. I don't think we take advantage of it to the degree in which we should. You talk about the pockets of clinical trial data that you have, and you can go back into it, and that's optimal and should be done when it can. We in the Department of Veteran Affairs, we are an enclosed system. We have longitudinal care. When our patients come in, they stay in. Not to say that they do not use other healthcare systems, but when you have a patient population where the disease state burden or the burden of illness is greater, they tend to use our system comprehensively. And also, if a disease state requires medications where the cost is exponential, they tend to use our system a lot more comprehensively. So to that end, when you look at a disease state like this, like amyloidosis, and again, I look at it from the drug side, not from the disease state side, but the natural history for patients that have been in the system can be obtained inside of a system like the Department of Veteran Affairs. Now, it may already be done. We have a big system, so it may be a study that's already done that I don't know of, but that would be a good way to look at it. It would also be a good way to identify a subpopulation if you're looking at the longevity of the disease and the disease state severity and when the patients get treated, what happens. So I think taking advantage of that would be optimal as well. So just a thought, and I wanted to, other thought I wanted to bring up, I could talk about, stay up here days and talk about real world, but I'm going to stop. The last thing I'm going to talk about really quickly is accelerated approval. So when you have accelerated approval and the drug begins to be used in the real world setting, how does a system assess that? We do begin to assess that because we're worried about our patients. We want to make sure that when the drug is being used, it is being used safe and appropriately. And so depending on how it comes into a system with prior authorization or with needing to do more criteria for use, typically we will track that very aggressively and have that data available to assess and to make available outside the system as well. We work collaboratively with the Food and Drug Administration in different areas with CEDAR, but I did want to put that out there because this is somewhere I think with this particular disease state, our patient population can be used a little bit more frequently.
Thanks for those comments, Matt, and then the gentleman there. So
I think we defined the problem, at length with respect to the three competing interests and the different potential trial designs. But I think it may be a good idea to get down to brass tacks and talk about surrogate endpoints or a combination of surrogate endpoints that could be reasonable. For example, a meaningful reduction in NT-proBNP, whatever that means, and a reduction, let's say, on ECV and MRI, or decreased hospitalizations and decrease in NT-proBNP, or some combination of 6-minute walk in NT-proBNP. I mean, we talked about the problem, but I'd love to hear about people's opinions and Dr. Stockbridge's opinions about would a combination of meaningful surrogate endpoints, at least meaningful to the patients and us, be reasonable versus, a mortality CV hospitalization endpoint with respect to approval or accelerated approval of a medication.
So you had a point, you had a comment to make?
Sure. Michele Mercury, I work in election, and I'm responsible for the amyloidosis franchise that we actually have. This morning, we spent a little bit of time talking about early disease and the change in the epidemiology of the disease. However, our patients brought back to the real unmet medical need, which is the moderate to severe patients who have been sparely studied and maybe not studied at all. Our patients understand the toxicity of amyloidosis, and I would say that amyloidosis is the equivalent of a metastatic disease. So we need to actually look at it as a cancer disease rather than a circulating protein. And I would say that, especially for some of the newest treatment, it is important to understand how to identify, quantitate, and prospectively monitor the amyloid deposit. And this, of course, is a challenge. I think that we have a few methodology and modality to identify the amyloid loads, but do not necessarily have the right data for prospective evaluation on whether we are actually able to eliminate this. Clearly, scintigraphy, CMR, PET, SPECT, we have a lot of different things, but they have not been necessarily utilized in order to prove the efficacy of a medicine. And maybe that's a territory and a topic that will need to be further discussed and further evaluated not in the not too long future. So thank you for your attention.
Thank you. John Burke has his hand up. Do you have a comment, John?
Yeah, very quickly. Julian Gilmore elegantly laid out the 5-year EPIC change in standard of care and its impact on the outcomes of patients who we're interested in with TTR amyloid cardiomyopathy. And so my question to the regulators on the panel and in the room is historical controls. How valid or how assured are we that they apply if we're talking about external placebos?
Okay, Norman, would you like to respond, or shall I ask LJ to also comment so that then you can respond to everybody together? Is that okay? So LJ, do you want to?
Yeah. Thank you very much. I think that's a very good question. I think using the real world data as a standard of care, and then supposedly we can run even a smaller trial or even one-arm trial, right? And how we can actually compare those two treatments. Of course, there are statistical methodology available, and James knows very well. For example, we call it a propensity score matching method. And we can also using regression, for example, you can use real data, you have outcome, right? Whatever outcome you want to use, and establish a relationship with a baseline variable to your outcome. Then you're going to move this model to your one-arm trial population, sort of like simulate the outcome if the patient actually in the one-arm trial population patient treated with standard care, how would the outcome look like? Okay, we did a several study like this one. We call this outcome regression analysis, which is very popular. And in fact, FDA utilized this methodology also approve a rare disease product in the past. So let me stop here. Let me later Norm make some comment.
Well, I think it is challenging to try and rely on a historical control in a setting where you think the natural history is evolving before your eyes. To the extent that you think you understand what aspects of the population are contributing to those changes in outcome, you can maybe design something where you can make reasonable decisions relying on historical control. And maybe the things that Dr. Wei is talking about address that. But if the treatment effect you find is huge, people will have much more confidence that that's a reliable finding than if you've got a barely statistically significant and relatively small treatment effect. Then everything you've assumed is at risk of having determined the outcome.
Yeah, Prem, if you don't mind, let me make a very quick comment. I think there's one issue about using real-world data. If we use a soft endpoint, for example, just 6 minutes walking distance, right? We don't really know in the historical data how the quality, how this performance of the 6 minutes walking distance. So it's not like a clinical trial with well-controlled situation, right? So we have this problem. If a soft endpoint, other than mortality, death is death. Doesn't really matter how you define it. And beyond that, even the cancer trial, we use a progression, right? Progression also have a different way to evaluate across the globe. So I think that's created some problem. So how do we actually guarantee your outcome measurement is really aligned with your clinical trial? So that's one issue.
May I make a quick comment on the topic of external controls? I think the situation in ATTR amyloidosis will be harder than usual for external controls. The changes in standard of care, the issue that was brought up about effort-based outcomes, these are all critical challenges. But what I wanted to emphasize is that we can be empirical and evidence-based about weighing these challenges. Ultimately, we would need to be confident that we're comparing apples and apples. What does that mean? It means we have to know what the important prognostic factors are and be confident that we know how to adjust for them when comparing one data source to another. We have to have confidence that we're comparing similar standards of care. I think that could depend on both time period and geography, as was brought up during the morning session. And we have to have confidence that the outcomes are behaving in a similar way. And so a simple way to assess that is we can take standard of care arms from different clinical trials and compare them to each other and compare them to real-world data that reflects the same standard of care. If those show consistent outcomes, that should give us hope that external controls could be feasible. If those are different, we should be very cautious about putting too much stock in external controls as a way of solving our clinical trial challenge.
Thank you, James. We've got a number of hands raised and the clock's running. So Dalia Dawood from NICE in the UK.
Yeah, thank you so much. Dalia Dawood, Associate Director from NICE in the UK. So I wanted just to come back quickly on the point of surrogate endpoints. It seems like in some areas it is really kind of inevitable that there will be a need for using surrogate endpoints. But I think that the most important thing is actually raising the bar for the evidence requirement to use these surrogate endpoints. We really need to be much stricter in terms of the validity of the surrogate relationship. For us as HTA agencies that look more at the clinical effectiveness side of the equation. So I think regulatory focuses on efficacy and health technology assessment, and that informs payer decisions, looks at effectiveness, so how well the treatment or the new drug is performing compared to current standard of care. So we need to assess that in light of multiple sources of uncertainty, and one key factor or source of uncertainty is the use of surrogate endpoints and how the surrogate endpoint is actually related to the final outcomes. In most cases, it just relies on epidemiological evidence, but there are levels of evidence, and the highest level of evidence is a meta-analysis of RCTs. So we have recently moved to requesting that we are provided with evidence of the validity of the surrogate when it's used in, for example, in the economic models as well, that there's evidence has to be according to these levels of evidence and to be highest level of evidence. So when we use economic models as well, we need to quantify the uncertainty. So it's not like a single point estimate of X change in the surrogate endpoint leads to Y change in the final outcome. But this relationship need to be also quantified in terms of what is the uncertainty in the relationship. And this need to be reflected if the surrogate is used in the economic models, then this need to be quantified in sensitivity analysis. So that's really important in terms of uncertainty, reflecting that there is uncertainty in this relationship, and we need to see this uncertainty because it affects our decision uncertainty. Thank you.
Thanks. We have about 10 minutes left. Adrian?
Just a quick, one clarification also, kind of what I hear as a summary for some of this is that, when we're talking about historical controls, again, thinking about that as very objective. And so like an oncology space, that can exist, or some other spaces, and it is typically a biomarker or a radiological finding that's used. And so I think it's something different than what some of the types of outcomes that we've used here. So it'd have to be something like that. The second thing is that it does seem like, we wouldn't be here if it was going to be the solution is a simple randomized clinical trial with placebo. The discussion would have ended about 6 hours ago. And it seems like we're going to be in a space where it's going to be a hybrid approach, taking advantage of where there are regions where it is suitable and ethical to do placebo-controlled trials with regions that may have a standard of care that approaches 100%, and being able to use a hybrid design for combining data so you can see what the so-called placebo effect is, but see what the effect is compared to some type of standard of control, standard where whether that's a randomized data or contemporary data.
Thank you. So, in all the talks, we heard about multiple approaches to using sort of composite endpoints, and whether we use that as a time to first event or as a cumulative area under the curve approach. In all of these approaches, we give equal weightage to all the components, right, of the composite endpoint, except in the win ratio approach, where you can actually say that death's more important than hospitalization. So in the example that LJ showed, you had that one case where you had 3 hospitalizations and the area under the curve was very big, and then somebody died very quickly, and the area under the curve was very small. So how important is it to treat, these different endpoints differently with different weightage? Matt, the ATTR Act was the first time we saw the win ratio applied, so maybe you can take that question.
I'll defer to LJ, but I think he has ways to figure out how to rank certain endpoints higher in his area under the curve. We weren't the first, actually. The partner trial and others used it. That wasn't really well advertised. It was pre-specified, but it makes intuitive sense. And I think when you're picking endpoints, we heard this earlier from Adrian, we want to be real, so we should sit down and think what matters most to the patient. And usually survival is important, assuming quality of life is okay, and then hospitalizations. I do think the win ratio is attractive. I also think there are deficits to it. It gets, too far down and too many hierarchies. I don't know what's happening when you're making a billion different comparisons. And obviously, it was very favorable in ATTR Act because we won on mortality, we won on hospitalizations, 2 hard endpoints. But LJ, maybe some thoughts about how to utilize that in your modeling?
Yeah, thank you very much. These are great questions. Regarding this very interesting question, suppose one arm has a short life, right, compared with the other one. And of course, artificially, you reduce the number of hospitalization. But if we're thinking about using a positive measure, for example, hospital-free survival, this concept, then think about it. If a patient with a short life, then automatically penalized the guys that have a short survive, right? So in that case, we handle both. You have this death involved, you have a hospitalization, and you say, well, 3 hospitalization equivalent to 1 death. The other guy said, no, how about 5? So there is no ending of debating for this kind of weighting stuff. Right? So I would say, thinking about a positive, desirable outcome, but with this really bad terminal event, and if a terminal event is short, then automatically you penalize this good outcome measurement, like a hospital number of days. But let me get a win ratio issue. Win ratio is an interesting idea, but on the other hand, win ratio got a problem in the sense that first, it's very hard to interpret. If your win ratio is 1.21, you say 21% increase. I say increase from what? It's like odds ratio or risk ratio without telling me the probability underlying, right? My probability is 0.01, increase to 0.02, its odds ratio is 2. I say, well, if you don't tell me 0.01, the baseline level, I have no idea clinically how to interpret. So win ratio the same way. We have no idea what do you mean by this so-called the underneath. What is the reference benchmark value? That's one thing. The other thing is the win ratio is a funny business. If you do a testing hypothesis, just getting p-value, I have no objection at all. But when you do the estimation, the size of a treatment, this is a funny business now. If you patient have a sensory data, then the win ratio estimating something involving sensory distribution. I said, well, gee, I'm not interested in sensory distribution. And how come, get involved? Unfortunately, that's a technical issue of a win ratio. But anyway, it's one of the technique, but it's good to get a p-value, but it's not good to get a measure of the size of the treatment.
Thank you. Ahmed Mashri.
Yes, hi everyone. I'm a cardiologist at Oregon Health and Science University. Just a quick comment on the CV hospitalizations angle. We as systems, we're actively working against this endpoint because we're employing more and more nurses since the COVID pandemic in order to, keep patients out of the hospital. And you might say, well, that's a good outcome. True, it's a good outcome, but it's actually against this outcome for clinical trials because some of the patients are seen once a week in the nurse clinic or even sometimes in a nurse practitioner clinic to keep them out of the hospital. Yet sometimes they can come to the hospital one time, get the therapy, and then get out of it. And one counts as an outcome, the other doesn't. And we've seen some trials starting to look at intensification of therapy or urgent or emergent use of outpatient, therapy centers. But all systems, including ours, are actively working against such an endpoint that we have been historically utilizing.
Thanks, Ahmed. And Clement?
Just one note on the win ratio. I agree with what has been said, that it may not be clinically interpretable. And if you want to have a clinical interpretation of the results, you have to go to the secondary endpoints and look what happens there. So it's difficult, but the regulators are flexible, and we have accepted that. And in combination with the recurrent event analysis in case of tafamidis, we're happy that we had not really made our decision based on that. I'm a little bit more skeptical regarding the recurrent event analysis than what has been proposed here. The EMA has made a lot of simulations together with the consortium or in exchange with the consortium that has been published in 2020. And the biostatisticians came to the conclusion that if you have a high mortality rate, then the effect on hospitalization will not become interpretable because mortality rate will have an effect on that. I'm a clinician, so that's beyond my understanding, but there are methodological issues. I'm more on the clinical side, and the assumption that was suggested in one of the slides, that the hospitalizations or rehospitalizations are equally distributed over time, may not hold true. What we see in the clinical trials is more of a clustering. If a patient has been hospitalized, then the chance to be rehospitalized in the near future is high, and we also see clustering before death as expected. So we have not equally distributed but clustered events. And then if you think about, does it really make a difference whether the patient dies at home or whether the patient goes to hospital 2 weeks before he dies? I'm not sure that counting 2 events in the second case versus 1 event in the first case really makes a difference regarding disease burden. So I'm a little bit more skeptical on the use of recurrent event analysis in the field of heart failure. Thank you.
Thank you. Charu?
Thank you. I'm Charu Gandotrab, clinical team lead in DCN. Just a few thoughts. I think a lot has been brought up about the possibility of doing placebo-controlled trials in the post-tafamidis world. And I'm wondering if potentially enrolling non-responders or patients who are intolerant to approved therapies might be one option. Also, if you're looking at maybe short-term trials that are looking at functional improvement or symptom improvement, maybe over a few months, maybe potentially there is, a feasibility to do placebo-controlled trials. Also wanted to just throw out the idea of platform trials, perhaps, if there are multiple therapies that are being evaluated, a potential benefit of having a single control arm and sort of building in sort of design where you can, see if there are promising therapies that can keep moving forward, or there is a potential early signal of questionable therapeutic benefit, the trials could be dropped. So understandably requires a large collaboration to be able to move something like that forward. Also, there was talk about trying to, figure out surrogate endpoints, and I'm wondering, within a registry setup, or maybe the VA system that was referred to, if you could plan something like a minimal intervention design where you are serially imaging patients. An example could be resolution of abnormal findings on P-by-P scan, and how does that translate into outcomes. It looks like a lot of work needs to be done. We don't have enough right now to, at least in the cardiac space, to figure out surrogate endpoints, whether it's BNP or maybe some imaging-based study. You want to, and Dalia nicely laid out, what is necessary when you're looking at magnitude of impact on a surrogate endpoint, and your sort of calculations considering uncertainty as to how it will relate to hard clinical endpoints. There was a question about using multiple surrogate endpoints. I think it's hard enough to figure out with a single surrogate endpoint, especially with this heterogeneous disease. And I think it becomes even more difficult if you're thinking about multiple surrogate endpoints. So,
thank you, Charu. Good points. We're a few seconds over time, so Matt gets the last word.
I just wanted to say, obviously, I think we all would like to look at the totality of the evidence. The one surrogate endpoint we didn't emphasize much, which I find intriguing in these patients that's emerging in the regular heart failure literature, is patients with amyloid develop over time lower stroke volumes, higher central venous pressures, lower cardiac outputs, and a syndrome of cardiorenal, and they need more and more diuretics. And the dose of diuretics has been shown in multiple databases to be prognostic. I'm not saying that's the only surrogate, but that's one I would encourage us as a community to try to explore, not only in the context of our databases, but in the context of trials, because it seems to predict bad events in patients over time.
Thanks, Matt. And I apologize. I know there were other people who wanted to make comments, but we are out of time, and I think just to be on time, we'll close this session and move on, and I'll hand over to Rick.
We have a break.
Oh, we do have a break. Sorry, we have a break. Yeah. Thanks to all the speakers for excellent talks and the lively discussion.
So welcome back. We're going to start the next session, which is diversity in research. And again, I'm happy to introduce the moderator, Rick Ruberg from Boston Medical Center.
Thank you very much, Dr. Maurer. It's my pleasure to introduce the president and CEO of the ARC, Isabel Lusada.
Thank you, Rick. Well, I have a in-depth one-slide presentation, so we can just pull up the next slide. Not that one. Maybe I can move forward. Hold on a minute. I can probably just move. Nope. As the slide comes up, I'll just get started, because I'm going to just briefly talk about the DEPICT Act, which creates the first ever national standard for diversity in clinical trials, with the aim of ensuring racial and ethnic minority groups are better represented in the study of experimental medicines. It mandates data reporting for large-stage studies on participant demographics. Sponsors will be required to report and to defend the rationale for enrollment targets by demographic. U.S. Representatives Brian Fitzpatrick of Pennsylvania, Ann Eshoo of California, and Robin Kelly of Illinois introduced the Diverse and Equitable Participation in Clinical Trials, DEPICT Act. This was actually passed at the end of last year as part of the 2023 Omnibus Spending Bill. So here we go. So the law which incorporates the DEPICT Act marks an important step in addressing decades of testing drugs primarily on white men. The practice left knowledge gaps on how drugs would work on a large swathe of the population. Black Americans count for about 13% of the U.S. population, and 22% of yearly cancer cases. But the median enrollment of Black Americans in clinical trials was about 4.5%, according to a study that was done in 2020. So what does the DEPICT Act? The DEPICT Act requires investigational new drugs and investigational device exemption, IDE, applicants to report clinical trial enrollment targets by demographic subgroups, including age, race, ethnicity, and sex, and provide a rationale for those targets. They need to provide a diversity action plan detailing the actions the sponsor will take, such as outreach and engagement strategies to reach these enrollment targets. The FDA has to produce guidance on diversity action plans for clinical studies, including enrollment and retention goals, rationale, and how sponsors will meet goals. It also requires that FDA publish an annual report aggregating and analyzing the data provided by sponsors on their progress towards and strategies for improving diversity in clinical trials. There needs to be a public workshop to enhance clinical study diversity, and also requires the FDA to hold a workshop to assess how sponsors utilize the clinical trial flexibilities initiated during the COVID-19 public health emergency and their impact on improving access to clinical trials in underserved populations. With that, I'll hand back to you, Rick.
Thank you very much, Isabel. So it's my pleasure to take us to the penultimate session this afternoon, building upon some of the discussion items that we launched upon this morning. If we can open up the Zoom, I will introduce our panel. So joining me in this panel, obviously, and with Isabel, is Dr. Alana Morris from Emory University. Joining us virtually, Kair Shah, here from Virginia Commonwealth, here in D.C. Dr. Mona Fuzat from Duke University, virtually. My friend Renee Bullock-Palmer from Deborah Heart and Vascular Center, Lung Center, sorry, in New Jersey, Thomas Jefferson University, and also Victoria Sanjurjo, sorry, here in Washington, D.C. So as I was organizing my thoughts around the discussion this afternoon, I was actually happy to see that slide, Isabel, that I had not seen yet about the DEPICT Act. And I'd like to kind of organize our discussion this afternoon around this issue of equitability and access. Around enrollment, number one, retention, number two, in other words, maintaining participants in clinical trials and creating trials such that people don't drop out or withdraw consent. And finally, collecting adequate data to determine whether the efficacy results that we attribute to the treatment are actually related to the treatment, or if they're not related, are not confounded by other aspects that we are not collecting, specifically determinants that potentially participants can't control. So moving to the first point regarding enrollment, I'd like to open the discussion, and perhaps I'll open it up to the panel first, regarding the thought of mandating specific enrollment targets for specific populations, the pluses and minuses associated with that. Matt mentioned this actually when he introduced the ATTR trial and how he was obligated to meet a specific enrollment and it resulted in closing enrollment to a certain population of people to meet that enrollment target. So I'd like to open that discussion and for comments. Anybody on the panel? Oh, Dr. Morris.
Yeah, it's one step in the right direction. I think without sort of pushing sponsors, site PIs, and other people who are involved in enrollment to target populations that have sort of historically been under-enrolled, how else will we at least sort of move the needle in that direction? There are certain populations, I think, that may be slightly easier to access, perhaps maybe have less sense of vulnerability or reluctance to participate in trials. And if we just go for the populations that we've always enrolled, we'll continue to get the same results that we've always gotten. And so I think that mandating this at least makes us as investigators and sponsors think about the issues at hand and have the conversations that we're having right now, which in the past, I think we haven't had with as much fervor as we're having right now.
Thank you. I agree with that. Comment, Renee.
Yeah, I do agree with Alana on that comment, and I think it's very important that we're obligated to enroll these study participants, and I think the number of enrollees, the targets should reflect the percentage of those populations within the community, and I think for amyloid, with it affecting the Black population so heavily, I think it's very important, and I think not just African American men, but also African American women as well, because I think within that population we are seeing this increasingly in momentum. Thanks.
Thank you, Dr. Fusat.
Hi, everybody. Great job pronouncing my name, by the way. Thank you. I just have a couple of comments. I'm representing today, I think, also the Heart Failure Collaboratory, which is a very similar consortium to the ARC. And one of the things that we've proposed is perhaps considering an incentive approach, meaning lobbying Congress to provide FDA the authority to incentivize sponsors similarly to the pediatric program, so that there is a great incentive from the sponsor side to enroll these patients and really get the information. But what strikes me with amyloid that is a bit different from heart failure in general is that the disease and the genetic variants track with race. And so it would seem to me that just by that scientific reasoning alone, there should be a strong incentive to enroll a certain number of those patients in the trials. So that's one thing that we've discussed is having an incentive approach. And then also another thing that we've worked on a bit is there's pretty clear data that the types of investigators that are enrolling in the trials do much better enrolling their own similarly, either Hispanic or Black or women, similar type of person in the trials. And so there's a trust issue. And so one of the things that the collaboratory has focused on is actually training the next generation of diverse investigators to also then attract a diverse population of study subjects or patients that enroll in the trial. So those are just a couple of thoughts that we've focused on with the collaboratory, the Heart Failure Collaboratory.
Thank you very much. I'm really glad you brought that up, because that was echoed in this morning's conversation regarding kind of onboarding and how do we actually bring study personnel up to the task of enrolling a diverse population. I'd like to ask maybe, Victoria, your thoughts from the side of a sponsor. I thought it was interesting also the way Mona brought up the concept of of an incentive rather than, say, a mandate. How the concept of incentivization, how you speak a little bit about the best approach to ensure adequate representation.
This comment is only from myself. I believe it should be a mandate. I do. I don't think unless you're forced to do something, I don't think anyone does it. And we've been working on this for 20 years, and it's still an issue. Heart failure is still the leading cause of Blacks in the United States. Hispanics are never mentioned in any decks. I remember when I started working in ATTR, we had a slide, it said 0% Hispanics. We know that's not true. Caribbean, Black Caribbeans, Brazilians, they're all under the Latinx umbrella. So as far as I'm concerned personally, yes, I believe it should be mandated. And once you mandate it, now that we know, now that industry knows that we have to do it, we will be doing it. Everyone's going to be doing it, because you can't get published if you don't do it, And if you don't have your plan, which is beautiful, then they're not going to let you move forward. Regulatory will not let you move forward. So I think it's a wonderful opportunity for us to actually advance health equity.
Wonderful. Thank you. Any comments potentially from our regulatory colleagues? They don't have to make them if they don't want to. Nobody's jumping in.
Go ahead. If there's time, I have
Sharmila, please.
Thank you. So I agree with all the points about mandating participation, But there's an entire another aspect of the participants actually willing to participate. Like, what are we doing as a community to encourage participation? And I can tell you that we are recruiting participants from the ARIC study from 4 different centers: Minnesota, Wake Forest, Mississippi, and Hopkins. And we have very little enrollment from the Mississippi site. For the same materials, we are trying very hard to bring them in. They're all in the bigger ARIC study. We just have very minimal recruitment. So one of the things we did, I don't know if it's I think changing a little bit, is when we looked at our brochures, we did not have any African American, either patient or a doctor. So the one thing we did was we changed the brochure, so we showed people like them, doctors like them. The second thing is the approach may have to be different, because a lot of these people are elderly and they're like, oh, we don't want to participate in the study because I've already lived my life, I'm happy. But what kind of resonated more with them was, oh, this might affect your family members. Maybe you don't personally benefit from it, but if we find something, it's possible that the next generation may benefit from it. So I think the strategies and approaches to reach this cohort may need to be developed further.
Thank you, Sharmila. I completely agree, and you've echoed many of the comments we made this morning regarding engaging trust and dissemination of information in a way that people can appreciate and digest and understand. I'd like to, in the time we have remaining, move to the next topic, and that is retention. How do we construct trials such that participants can follow study procedures and follow through so they can achieve either efficacy of therapy or not, depending upon what they're randomized to. So maybe I'll ask Dr. Shah, can you comment on that? Would you like to comment on that?
Yeah, sure. And I want to follow up on some of the other comments earlier.
Please do.
Okay. So I completely agree with all of the speakers. I think Alana made a point that this was a good first step. And it does. It kind of addresses one end of the spectrum that we should be discussing, and that's policy and institution. And then I completely agree that the focus from our end should be on patient interaction, language, conversation, cultural identity. And I think it comes back to what Dr. Ruberg and Mars pointed out earlier in their most recent study is they met with patients, with patient advocates, the groups, to design the study. And I think if you start there and you hear what they would like answered and what they would participate in, depending on whoever your target study population is, I think it'd make most of the execution and retention a lot easier, and engagement a lot easier. But that being said, coming back to your original question to me about retention, I do think if in my short career, short relative, I've found that the more recent studies with amyloidosis have really addressed a lot of the retention issues, challenges I had earlier on, in that I think the investigators are sensitive to, in this elderly population, things like transportation, home visits. All of that has made a huge difference in at least our site's dropout rate. I'll say one more thing about enrollment, and specifically enrollment of V142I patients, is we've kind of done this to ourselves, the way we've designed these studies. We have combined studies where we treat wild type and variants as the same disease, and certainly there's a lot of overlap, but you have a prevalence of one which is just drowning out the other, not giving us an opportunity to understand what's going on with these drugs in a population we're trying to find blame in for why we're not enrolling.
Thank you very much. Great points, Dr. Shah. Dr. Bullock-Palmer.
Yeah, I think we have to, can you hear me?
Yes, we can hear you.
Okay. But I think as we're talking about retention, I think we have to meet patients where they are. And, it's once we have these patients enrolled, the challenge is keeping them enrolled. And, I think reimagining the studies in terms of, not having it centered around the major healthcare centers, but then probably having, dispersing the study workforce in local community health centers, engaging the primary care physicians who are having these frequent visits with the enrolled patients so that they themselves understand the study so that they can answer questions of the enrolled patient, and also having flexible enrollment hours, because many of our patients do not have a 9-to-5 job. Some of them may have multiple jobs, do not have a reliable transportation. So having ways in which we can have the study sites being closer within these communities so that it's not too much of a barrier for them to actually visit these study sites. And sometimes even there are financial barriers, because if they're missing certain hours of work, that's less pay for these patients. So probably if there's a way to incentivize the enrolled patients to participate in the study. And I think having frequent town hall meetings, once these patients are enrolled within these various communities, having a Q&A session, town hall sessions, which could even be virtual if possible, or live, so that patients can have their questions and concerns answered as they're going along in the test, because in the study, because there's a great sense of mistrust, and I think trying to dispel some of those barriers would be important. Thanks.
Thank you, Renee. Dr. Keith Ferdinand this morning made similar points when we discussed the concept of community partnerships and advisory boards. So I think those are definitely of critical importance. Dr. Burke, you'd like to make a comment?
I think this may be actually a question for you, Dr. Ruberg, and that is, at Boston University, our process is to try and encourage people who come through the Amyloid Center to participate in clinical trial. And while I applaud and embrace the issues of retention and mandated enrollments, it strikes me that where we fall down is actually identifying the African American population that's affected by disease. We just aren't diagnosing the tip of the iceberg. And in order to get people in clinical trial, they have to know they have disease. And so I think the SCAN-MP study has certainly enhanced our interaction with the African American population in the amyloid clinic. And Dr. Ruberg, what recommendations do you have for the group about how we enrich our interactions with the underrepresented?
Well, thank you, and I will tag my colleague in SCAN-MP, Dr. Maurer. But I would say many of the things we've been discussing already are the ways in which we constructed the study: advisory boards, community partnerships, and constructing the study in such a way that we thought about it from a patient experience and to make it something that patients could do. It was not particularly onerous, and also that had as minimum inherent biases as possible, so that we were able to get the most people to diagnostic testing and identify as many people as possible. I think the issue of bias is really important, and that was something that Victoria brought up earlier this morning. Bias to ordering appropriate testing to recognize disease, bias to referring to clinical trial. These are all problems that we need to kind of confront and try to overcome. I think that's how I would answer it. Matt, do you have any additional comments you'd want to make?
Yeah, I guess I would say, John, that I applaud the NIH for recognizing this is an unmet need and they need to invest some resources. I think we all have to think about what resources we need to invest, whether we need, as we said, programs in universities with historically black colleges or places where patients actually reside that are disproportionately afflicted by this condition. I just point out that in SCAN-MP we have 4 sites: Harlem Hospital, Columbia, Yale, and Boston Medical Center. Harlem is fantastic, and they're fantastic in that literally 50% of the patients that they find who are eligible, they enroll. At Columbia, that's not higher than 25%. And I think their mechanism of doing that is because they sit down and they talk to the patient face-to-face, and we have this idea that we're going to call people on the phone who we've never met and tell them, you can't pronounce the word amyloid, that you have a life-threatening disease, but we're offering you to come in and get tested. It's just a hard conversation to have. And so I think we have to think about ways to engender their primary care provider and other people who they trust before we're going to get anyone to agree to, get further evaluations.
Thank you, Matt. I'd like to recognize Dr. Vaishali Sanchowala from Boston, and seeing you on the Zoom is almost like we're in one of our clinical meetings for me. But Vaishali, you have some comments to make?
Yeah, so, again, I think I just wanted to make one comment, and that was about the Aquarius clinical trial, which is a clinical trial for AL amyloidosis, done as a follow-up trial to Andromeda clinical trial mandated by the FDA for J&J or Janssen. And this trial is a deferred administration of Cyborg-D in patients with AL amyloidosis. But they have also mandated that there are 30 patients, cohort B or cohort 2 requires 30 patients to be enrolled on the trial who are of minority, ethnic minority or racial minorities. And of those, 15 have to be African American. So, again, I think that the FDA or the regulatory agency has mandated, and now Janssen has really engaged organizations that educate, engage, and empower patients in the Black community and minority community. They have connections in the churches, in their community health centers, where these organizations, they know people, or I don't really know how they do it, but they are able to. The unfortunate part is that if people do have amyloidosis, they know that these trials are available. But as Dr. Burke said, that it's not, they're not going to be able to diagnose themselves, on their own. But, these efforts are ongoing, and I think we are much better now than we were 5 years ago or a decade ago.
Thank you very much, Vaishali. As Alana said, it's the first step to moving the needle, I think, and so this is an important thing to consider. I'd like to actually.
Can I just jump in with a quick comment?
Of course, please.
I think there's a theme that's emerging here. We can talk about how we're not doing enough, et cetera. But I had an accounting professor who said, life is just incentive compatible. People do what you give incentives for. And you've heard multiple people here say, we probably need financial and non-financial incentives coming from our regulatory agencies to say we won't accept data where your trial population doesn't look like the actual population you're going to use the drug in. Because the truth is, as Matt said and Victoria and others have said before, that this is a resource-intensive endeavor. We don't want to minimize the issue that if we want our trial population to look like the disease population, it is going to take resources, whether it's redesigning materials, developing new partnerships, or investing in the community. But for those resources to actually be allocated, we're going to have to provide material incentives, and they may be financial or non-financial.
That's an excellent point, Dr. Kazi. Thank you for making that. Not only can we could mandate, but we also have to provide the tools to be able to meet that mandate. I agree with that. In the few minutes remaining, I wanted to turn back to the panel and talk a little bit about what should we measure in trials to assure that we are understanding potential confounders? And this gets to the concept of the, for lack of a better term, social determinants. And it's a very large topic, but I thought I might ask the group, maybe start with Alana, what do you think we should be collecting? What sort of information so that we can begin to understand if, say, a treatment is negative, it was not confounded by something we didn't actually measure or think to measure?
Sure. I mean, I think in general, we probably don't think to measure most of the social determinants prospectively, and that's been, I think, a real challenge for us in cardiology, right? We're often doing geospatial analyses based on zip code that's collected after the fact. Half the population is missing the data. We're using sort of census level metrics as opposed to individual level metrics, which we know that there's often a strong correlation between individual level and census level SDOH, but they're not the same. And so I think if we want to tackle some of these questions, we have to collect this data prospectively. Now, of course, sort of this brings up the question of burden, right? Because within these trials, we're often interested in collecting biomarkers and, adherence data and the med log and, vitals and the baseline stress echo, rest echo. And then now you want to add in 16 questionnaires related to stress and social support and education and all of the things. But I think we also have to, when we're thinking about designing these trials, identify what are the important areas that we want to look at. I remember writing an editorial for an analysis of GUIDED that looked at sort of the hospitalization rate in Black patients in the US compared to all of the other sites, and the rate of hospitalization for those patients, despite actually getting more GDMT than the other patients, was higher. And so the assumption was that this was related to social determinants of health, but we don't know that if we haven't measured those confounders and collected the data. So again, this is a study design question, it's a study design issue, and we have to think about it as we're putting the study together as opposed to retrospectively.
Excellent points. Thank you. I think your point also is really well taken about the burden to participants. I know in ScanMP we thought a lot about that, and we focused on a couple of things we thought we could get done, like health literacy and et cetera, things that potentially also were modifiable. Maybe I could ask Mona, does the Heart Failure Collaboratory think about this? This is something that would seem to be not applicable only to ATTR amyloidosis studies, obviously.
Yes, absolutely. And in fact, one of the projects that we've undertaken is to create a core case report form. We call it a lean CRF, but Dr. Stockbridge does not like the term lean. So we're just calling it a core set of data that should be collected in every trial. And this year we're going to be creating a module for collecting social determinants. So that should be shareable and available to the public for use. But that's certainly something that we've identified as well as being very important. I think, as Alana mentioned, we're really mostly doing analyses by zip code, and very few trials even collect that information. So it's really not been very accurate.
Right. Agreed. I think looking at zip code, area deprivation index, those sorts of things are, I think about them as there are things that are difficult for us to modify. Like we can't really modify the built environment. We can try to impact things like food insecurity and housing insecurity, but that's out of the purview of a clinical trial. That's a bigger picture item, obviously. But things like health literacy, or things that like Dr. Dorbala mentioned about education or trust in the system and engagement, those are things I think we potentially can modify. Maybe not in the context of a clinical trial, but in the way in which we actually deliver medical care and offer trials to all of our patients in kind of in an unbiased, equitable sort of way.
Yeah, if I could just make one, I was going to just add one additional challenge, and I think Rosalind Adigan, who's there, has been working on this as well. One of the challenges, I think, for the FDA as well as others is defining racial groups. When we did go to create the lean CRF, our core group of demographics turned into a list of 12 or 15 different types. So just saying you're Black, and now there's gender versus sex and how you identify, is it what you're born with? So all of these kinds of issues are getting more and more complex, and the population is getting more diverse. And so, just the self-reliance on reporting your own race is also creating an additional complexity. So I think it's a big challenge for the FDA to define what those groups actually are.
Great points. Did you want to make a point, Rosalind?
Yes, thank you. Thanks, Mona. So just a quick plug-in. There will be a public workshop November 29th and 30th this year, where we're going to be engaging the public about what these diversity action plans look like, the challenges, and there will be an opportunity to get feedback from everyone. I think everyone here is a stakeholder, so I would just say be on the lookout. It will be a great opportunity for further discussion on this very important topic. Thank you.
That is a perfect ending. We've reached time. Oh, is there one more? I'm so sorry, Deirdre. Sorry, Dr. Mooney.
I'm reading some of the Q&A, and one of the questions, two questions came up. One was, do we have any data on the Latinx or Hispanic population with the prevalence of any type of amyloid, particularly ATTR? And then another one was, I live in an area where there isn't a center of excellence. What can and somebody who is getting treatment with a TTR silencer with a known mutation, and they were saying, how do I find a trial for myself? So if anyone could speak to that.
Yeah, so there is data. There was a paper published by the Mount Sinai and Penn group about, Caribbean patients who are affected. At Columbia, we have a Northern Manhattan aging study in which we have 800 Dominicans, and we've genotyped them, and the allele frequency of V122I is 0.88. So it's less than in individuals who self-identify as Black or from West African ancestry, but it's not insignificant. It's about 1%. And clinicaltrials.gov provides patients access to all ongoing clinical trials. And obviously, if patients are interested in participating, they can reach out to any of the centers that are listed there.
But I would add, and thank you, Matt, to that point, Deirdre, you do underscore another inequity, and that is regional inequity, where parts of the country have better access to care than others, particularly rural parts of the country without academic medical centers, which is another, I think, challenge for all of us. So we are a bit over time. This is a great discussion, and I'll pass it back to Dr. Maher. Thank you very much.
Thanks. So we're going to head into our last session, saving the best for last. We're going to talk a lot about access. This is a session titled Clinical Trials, Drug Development and Access. And we have two presentations, one by Sharmila Dorbola from Harvard and Brigham Women's and Drew Kazi. So I'll turn it over to them.
Thank you so much, Matt. Thank you all. Hi, Sharmila Dorbola from Brigham and Women's in Boston. So I've been tasked to talk about epidemiology of this disease. So I just wanted to share some existing data on the topic. None of the existing data are perfect, but this is what we have and something to share and discuss. So one of the publications was from Dr. Kevin Alexander when he was at Brigham, and he looked at the CDC death certificate data. And this was from 1979 to 2015. This included over 30,000 individuals. And key points in this. So look at the bar graphs on the left side, where you have age-adjusted death rates for amyloidosis reported on the death certificate. And there you have the race and sex, women and men. And you can clearly see that the bars for African American women and men are much higher. Age-adjusted mortality with amyloidosis is much higher in those two bars compared to the rest of the groups. And again, when you come to the right side, you see the map of United States and various different states there. And the bright pink color indicates higher reporting, and the blue color indicates lower reporting. And here you can see that there's a higher reporting of amyloidosis on death certificates in Minnesota and in the Midwest and in the Northeast. And one of the takeaway points from this is, this is just reporting on death certificate. So this may be a reflection of knowledge about the disease, evaluation of the disease. It really doesn't tell you that people in the Midwest are more predisposed to this condition or not, but it's more about geographic differences in thinking about amyloidosis. So if you didn't think about it, it doesn't go onto the death certificate. So this is an area that gave us some indication that maybe there's some differences, and perhaps centers which are closer to expert centers with knowledge about amyloidosis may have better reporting of this disease. Then we went ahead and looked at the fee-for-service Medicare data in the United States, and Lauren Gilstrap led this work, and looked at hospitalized patients over the age of 65 with an ICD code for heart failure and an ICD code for amyloidosis, and looked at incidence and prevalence rates. And you can see the incidence rate from 2000 to 2012 is going up only slightly, but the prevalence rate is rising. And I'm not showing you data here, but clearly these numbers were much higher. The prevalence rate was rising in older individuals, older men, and more so in African American men and in women. So what does this represent? Again, we can't be very sure because this is group data from a large number of people, but what we believe is that around 2005 is when the MRI findings were reported for amyloidosis, and after that you see the use of PYP and all. So it's possible that we have a lead time bias in diagnosis of this disease, therefore people are living longer. So remember, this is pre-tafamidis, pre-treatment for TTR amyloid. So it's possible that improved diagnostics led to lead time bias, and therefore the prevalence was higher. But what was interesting for us was if you look at the maps from 2000 to 2012, all parts of the country had higher prevalence in 2012 compared to 2000, which is good. But look at the 2012 map. Again, the Midwest and the Northeast, this is almost like a superimposable graph compared to the death certificate, suggesting that from two different data points, one death certificate, two hospitalizations for heart failure with amyloidosis and heart failure codes, we're getting very similar information, which is the Midwest and Northeast seems to be recognizing this condition more, coding for this disease more compared to the rest of the country. The Southeast portion of the country, as someone pointed out earlier today, is where we see greater proportion of individuals of African American background, and we are not seeing as much reporting in that area. This is old data, more than 10 years. This needs to be looked at, I think, in the more recent decade. But again, more work needs to be done in this area. So what are the modern trends? I think the modern trends is that data from the TEOS registry, of which Matt Moore, Dr. Pablo Garcia, and others were part of, looked at 1,069 patients and showed that we are now diagnosing this condition more with bone scintigraphy, shown in the orange bars, than with tissue biopsy. And this could be accounting for the lead time bias and greater detection of earlier disease, and this is something that you had heard of from multiple investigators earlier today. What other data do we have? We have data from Dr. Pablo Garcia's group, looking at patients who have HFpEF, who have thick ventricles, who are hospitalized and or elderly, showing that about 13% of them have DPD positivity. Then surgical aortic stenosis patients, about 6% of them have DPD positivity. Data from Dr. Matt Maher's group, patients undergoing TAVR for severe aortic stenosis, nearly 16.6%. Data from the Mayo Clinic looking at community patients with HFpEF older than 60 years of age, thick ventricles. They showed that about 10% of the males and maybe 2.2% of the females, on an average 6% or so, have PYP positivity, suggesting that they may have undiagnosed disease. And the screening cohort did better than the actual disease cohort. And of course, emerging data with carpal tunnel surgery, two different studies, pointing out that about 8% to 9% of the patients who've had previous carpal tunnel surgery do manifest evidence of cardiac involvement by bone avid tracer scintigraphy. Again, if you take all of these together, I think the prevalence of this condition varies from group to group, but it is safe to say that there are many different red flags we could be looking at. There could be significant underdiagnosis of this condition, and because we now have treatments to stabilize this disease, it's very, very important to look for this. This data has been shown already, the UK NAC data showing that the temporal trends showing that we are detecting earlier disease states in the more recent cohort, 2017 to 2021, compared to the earlier cohorts. And this was also shown by at least two different presenters, showing that the more recent cohorts, the orange bar on the top, tend to have better survival compared to the previous cohorts. Very similar results also shown by a Danish nationwide study. Again, everything pointing out that the epidemiology of the disease is changing. It's seriously underdiagnosed. It is much more prevalent than we are recognizing it clinically. And if we can detect a disease early, these patients seem to have better survival. And whether this is lead time or soon, we may be seeing effects of treatment as well. So let me stop there and take questions.
Maybe we'll ask Dr. Kazi to present, and then we can open it up.
Good afternoon, everyone. I'm Dhruv Kazi. I'm a cardiologist and health economist based at Beth Israel Deaconess in Boston. Thank you. And over the next 5 or 6 minutes, I want to walk us through the economics of both ATTR in general, but also specifically ATTR therapies. I realize that much of what I'm saying will be very familiar to this audience, and so I'll allow room for questions. I have no disclosures. So in the spirit of discussion, I will say that the Orphan Drug Act has been a victim of its own success. Since its approval, it provides strong financial and non-financial incentives for therapies for rare diseases, which in the U.S. is defined as conditions affecting fewer than 200,000 individuals. In Europe, it's about the definition is 1 per 2,000 individuals, which is about the same. And while each of these diseases is rare, collectively, the estimates suggest that they affect about 30 million Americans. So collectively, it's not a rare set of diseases. There are about 7,000 rare diseases, we think. And in recent years, more than half of all drugs that are approved are for rare diseases. So that's ranged from 58% to 52% in the last 3 years. And what has happened here is that we've set up an innovation access trade-off. On the one hand, these breeze through the regulatory process, and I'm being hyperbolic here by using the verb breeze. I mean, there's a lot of work that goes into them, but they get through the regulatory process, and then they enter the market at very high launch prices. So, whether we like it or not, tafamidis is the poster child of this problem, that it was approved by the U.S. FDA under the Orphan Drug Act, and then entered the market at $225,000 per year, which at that time was the most expensive cardiovascular therapy, oral cardiovascular therapy. And this has implications for all of the stakeholders. So let's walk through the implications for stakeholders. For manufacturers, of course, this has strong financial incentives, and this is seen in the fact that orphan drugs are attracting a lot of research investments, the fact that 50% of drugs are now in that space. There may be evidentiary thresholds that are adapted based on the feasibility. I mean, you can't run a 10,000-person trial, so let's run a 50-person trial. What can we get out of that trial, et cetera? But really, there are economic incentives. We talked about these high launch prices, and but there's also, we want to remember that the moment a company gets an orphan drug designation for one of its molecules, there's a 3 to 9% increase in its market value. So smaller companies see a 9% increase in the market value, which is probably commensurate with the downstream value of this molecule or this asset. But they also retain market power. So we think about this, that, nobody in the U.S. pays market price. They're all discounted. That's actually not true for orphan drugs. That because they have so much market power, by definition, they have no competition. The rebates are in the 5% range. And we talked about how the market recognizes the value of this by giving them a 3 to 9% increase in the capitation, in the value of that company. The Inflation Reduction Act, and I'll show you some data, is the most significant change to how Medicare is going to negotiate prices with for medications. But it has a rare drug exception. So if something has an orphan drug, a rare disease exception, so if there's an orphan drug, that is off the table. Prices cannot be negotiated on orphan drugs. Here's some quick data. Over the last 12, 15 years or so, the launch price, the price at which a drug enters the market, has gone up from $2,000 to $180,000. More than half the drugs that enter the U.S. market today enter at a greater than $150,000 per year, and this is driven by orphan drugs. And then, we've had some success convincing payers and manufacturers modulate their price based on some metric of value. So value-based pricing is going to increasingly become a new reality. As someone who lives in this space, I will say that it is not the reality for orphan drugs, and that goes back to this idea that there's so much market power that comes with having an orphan drug designation that even when there are studies that show that this is a hugely overpriced drug based on the value it adds, there's very little incentive to reduce the price of the drug. And an example here would be that we looked at tafamidis, and we built a disease simulation model, and Matt was part of this project and was hugely helpful in how we built the model. We calibrated it using trial registry data, showed that it actually worked really well, and here are the top-line results, that it's actually a very effective drug. So if you think about this, a 2-year improvement in survival is huge for a drug, and 1.29 QALYs. To give you an example, would be like coronary disease drugs add like 0.05 QALYs. 0.2 QALYs would be huge. So 1.3 QALYs is a really big change. This is a very effective drug, and I suspect that now in hindsight, that model probably underestimated the benefits of this drug. But even so, you cannot offset the fact that the drug costs $200,000 a year And so you have an incremental cost effectiveness threshold of about $900,000 per QALY. Now, to put this in context, the U.S. empirically thinks that the threshold is around $100,000 and $150,000. You want to come under $100,000 and $150,000 per QALY. I think for rare diseases, that's an unfair target. I think we need a higher threshold. But let's say we said $250,000 per QALY for rare diseases. This is still very expensive as cost effectiveness thresholds go. For us as clinicians and for patients, we heard really powerful stories today, and I want to thank the patients who shared their story. This means that we have more safe and effective options, but it also means that there are higher out-of-pocket costs for some of these therapies. I'll show you some more data on this. So in 2022, a prototypical Part D beneficiary could expect to pay $15,000 in out-of-pocket costs. Now, I had a statistics professor who said, statistics is the science that says if your head is on ice and your feet are on fire, then on average you're quite comfortable. And I will caution you that this $15,000 number is in that range. I mean, there are patients who will pay nothing, and in fact, we think that about half the patients pay nothing, and the other half pay quite a lot and basically forego the therapy altogether. But here's what's about to come down the pike. The Inflation Reduction Act, while it does not touch the pricing of rare disease drugs, it does protect patients from out-of-pocket costs. So Part D beneficiaries who currently can expect to pay about $15,000 per year for tafamidis will see, starting in 2024, a dramatic decline in their out-of-pocket costs for tafamidis. And that it relates to two different clauses, but most importantly, the elimination of the catastrophic coverage coinsurance piece, as well as the maximum out-of-pocket cost of $2,000. So basically, everybody's going to pay about $2,000 for this drug. It remains to be seen what this does to our patients. I would suspect that it will dramatically improve access to these therapies. We'll also make it possible to have combination therapies because you cannot pay more than $2,000 beyond this if you can get them approved. What does that look in terms of savings? If you just look at the last row there, cardiac amyloidosis, a prototypical patient under the standard Part D benefit will see their out-of-pocket costs go down from $15,000 to $2,000. I'm hoping that means positive things for access. But of course, someone has to pick up this tab. It's not that the drug has become cheaper, and they're not going to do so willingly. And so we're going to see pushback from payers. And I'm already hearing conversations where it's almost easier to get a drug approved than to get it paid for. And how payers are going to respond is they're going to be concerned about budget impact. And so there's going to be formulary restrictions, prior authorizations, and for the commercial side, meaning people who are not on Medicare, higher out-of-pocket costs. So it's not a clean story yet as to how the market will respond to these huge changes. I suspect the Inflation Reduction Act will do a lot of good, but there will be unintended consequences that we need to monitor for. And so where do we go from here? I mean, I think the economics of this is really powerful, and we need to pay attention, both as patients, providers, and health systems. We need to de-risk innovation costs. Can we create a system by which we reduce the cost of innovation in rare diseases? And Dr. Gandotra had an earlier idea. Can we build a platform by which we can test these quickly and so that people can fail, so trials can fail fast if they're not working, but we can divert our patients to the most successful trials? I'm a big believer on economic incentives to get people to do what you want to do. So tying regulatory incentives to downstream access is something that we have to think about. Value-based payments models are going to arrive, whether we like them or not. It's hard to see how they will apply to rare diseases because there is so much market power, as I mentioned. And thinking about what it does to patients in the commercial market. For amyloidosis is different because most of our patients are Part D beneficiaries in the U.S. and therefore will benefit from the out-of-pocket cost cap, but we'll have to see what it does to the commercial beneficiaries. All right, that's all I got.
So thank you both. Maybe I'll start with, if I could, Fran Cunningham, who represents the VA, where obviously there are a lot of older men who are at high risk for this disease, but you're in a, I assume, somewhat capitated system, right? So maybe you can describe to us any difficulties or challenges that you've experienced in managing tafamidis access. I would say parenthetically, I'm very grateful because I have several veterans who've gotten their drug from the VA at no cost, but just curious what's going on there.
Sure, no problem. So in our system, regardless of the drug, a very prevalent drug from a very prevalent disease state, or an orphan drug, or from a disease state that is not as common, we have to assess the drug the same regardless. And so every drug that comes into our system goes through the exact same evaluation, a full evidence-based evaluation with a fair amount of review, both internally and then throughout the VA system with subject matter experts, as well as those that would be prescribing it if they're primary care and it's not a subject matter expert agent. And then it comes back to a formulary committee, and then it's assessed and voted on at that point in time. And so for this particular agent, or agents like this agent, this one is on formulary with prior authorization. And so with that prior authorization, there's a set of criteria for use that has to be adjudicated for every patient that can potentially get this agent. And so we have a fair amount of patients that are getting the agent, and so it's very costly. So what happens? There is a give and take, of course, from the providers and patients versus the visions or whatnot that are responsible for paying. And so it comes back to, what are we going to do with this? Because it's taking a lot of, it takes up a lot of funding. And so there remains a lot of discussion, but nonetheless, our veterans need the drug, and so they get it because they go through a prior authorization accordingly. I can tell you where things become important to look at, and that's where evidence may not be as strong. Okay, where you see certain things, but the evidence may not be as strong. A certain dose. Is that dose, the study where the dose was defined, was that a sound study? Was the evidence really that good? And so in those instances, we have to take our own data and begin to look at it. And so that's what we're beginning to look at now. If we look at the difference in doses over time with our patients and compare that cohort and look at certain outcomes, look at mortality, look at cardiovascular hospitalizations or heart failure hospitalizations, to begin to do a comparison to say, okay, yes, this dose should always be started versus another. So they don't know if that helps or hurts, but that's kind of our world.
I think those insights are great. I know practically MedI and others who prescribe this medicine all the time rely very, very heavily on specialty pharmacists. We have two representatives here, Lynette and Megan, and I think it would be helpful for maybe you to describe, briefly, what do you actually do to support patients and how much time it takes? What are the stress points for patients and for the pharmacists who are dealing with getting access to this medicine or these medicines? Either Lynette or Megan.
Yeah, sure, I can start. So at Boston Medical Center, we have a specifically embedded specialty pharmacy within our site, right within our clinic. We have a specific dedicated pharmacy liaison and pharmacists who specifically handle prior authorizations, access, and financial assistance for our amyloid patients, most often being this with tafamidis. This process generally starts once we're notified by the amyloid physician, so that early conversation is important to involve pharmacy from the beginning once a treatment decision has been made. There's a lot of behind the scenes efforts that are being made before the patient can even obtain the medication. So once our pharmacy team does obtain the PA approval, most of the time this is associated with exponentially high copays. The large majority of our patients are from underserved and minority patient populations who oftentimes cannot afford these high copays. So for that reason, we often utilize patient assistance programs for our patients, for those that do qualify. But even though there still are many barriers and delays that can impact our patients' access to the medication, and that can hinder the start of therapy. So patient assistance programs do require manual paperwork, financial information, and signatures for our patients. So there are many challenges in those specific patients with lower health literacy. And because of that can start the delay of the approval for the patient assistance program and ultimately the start of treatment. In many cases, this can be delayed even by a few months. Just the other day, sending a prescription for a patient that was intended to start therapy. We had prior authorization approval. 3 months later is when we're starting the medication. So at Boston Medical Center, we have made a lot of efforts at our institution to help mitigate these barriers. One way being so that we developed our own internal patient assistance program with a little bit more simplified and expedited review process. And this, being to help reduce those delays for those qualified patients. We do, like I mentioned, have pharmacy liaisons who will walk through all the information with our patients and go through any financial paperwork that's necessary. We do have access to 24/7 translator services for patients who have different language barriers. When we have access to a variety of languages. And we also have Spanish-speaking liaisons within our pharmacy. And they do have access to clinical pharmacists. So I am on site in the clinic near the specialty pharmacy. We have multiple clinical pharmacists available for teaching, education as needed. And these pharmacists are available on call 24/7 for any emergent patient calls.
Thanks. Very helpful, Megan. Things to add? I'm just curious, maybe someone could comment to you. How many hours do you spend per patient per year in dealing with these issues?
Yeah, just to piggyback off of what Litnet was saying, we have the same kind of procedure at our Shields Health Solutions, the company that I work for, work directly with NYP Specialty Pharmacy. As per hours per year, I would say 25, 30 hours a week. So whatever that is multiplied by 52 weeks. Our liaisons, that pretty much is their dedication to their job, to their patients. That's what they spend most of their time doing. If they're not getting financial assistance, they're working on prior authorizations, trying to get the patients free medication. So it's a tireless job for them, but they do it very well. And I know the patients are very thankful for them because they wouldn't even know where to begin doing this for themselves. So it's something that the patients definitely need on their side. It's a tireless job for our liaisons for sure.
Thanks. Maybe I could ask Dahlia and Martin. I know, Dahlia, you're obviously with NICE, and you weren't involved, but decided not to initially approve tafamidis. My sense is mainly for this issue of value. And Martin, you were previously, I guess, a consultant or part of NICE system. So it'd be helpful to understand because, I don't know, it seems like in the U.S. it's whatever the market will bear, and that doesn't seem, as we heard, so rational from Dr. Kazi. So any thoughts you might have in this particular regard? It seems like we're on a unsustainable train that's going to crash at some point.
I don't know. Shall I jump in first, Dahlia, and you can give
sure.
current NICE position. So I was a director of NICE on the board for 4 years and been a cardiologist for 25. And it's only in the last 12 months I crossed to work full-time with AstraZeneca on our amyloid programs, closely with Iona's colleagues and Michele from Alexion Rare Disease. So a broader perspective on that. My observation of that is that it's really important the narrative from patients and clinicians lines up with the company position. This is about selling a value story to a reimbursement authority. And I think listening to all of the really excellent discussion today, it strikes me that we have to get the balance. There's no point trying to do it faster and cheaper and then not persuading either the regulator or the payer community. If the payer community is not prepared for the discussion, doesn't understand the disease or the impact on patients, or we've not measured it properly, or we just don't articulate the story well enough, then you lead to problems. And I really liked Michele's comment about thinking of amyloid in a more oncological way. Patients present already with advanced disease, lots of amyloid. This is a really critical situation. This is not just a chronic condition of cardiovascular disease. So trying to change that narrative is really important. And to Dahlia's earlier point, there is a lot of uncertainty here. The trials are short, they're not very large. Often the endpoints are not conventional. So how do you deal with the uncertainty when you try and project for the whole lifetime of that patient? So I think I'm not giving the answer to this, I'm just giving the context and the landscape that we have to get better as patients and clinicians and drug developers in landing this properly. But it's going to get more difficult because it's going to end up with combination therapies for patients. What sequence, what drugs, when, for which patients, when do you stop? Those are things I think we as a community need to sort out. And at that point, I'm going to hand back to Dalia, who can give you more contemporary, nice input.
Thanks, Martin. So just a disclaimer first that I'm not representing NICE views here, and I wasn't involved in this specific appraisal. But exactly as Martin said, it boils back to the uncertainty about the long-term extrapolation of the benefit beyond the trial time horizon, and that's something that is a major driver of cost effectiveness in the longer term. So this was a major issue in this appraisal, as far as I understood. And it's exactly about the fact that when you extrapolate for a long time horizon, you are making a lot of assumptions about what happens after the drug is stopped, and what are there patients that continue on the drug but don't benefit, what is the cost involved. So there are multiple factors that then come into play when you start extrapolating for a lifetime horizon when modeling and assessing cost effectiveness. So this is really a key issue here with these drugs, and I think we've discussed about the potential for improved for understanding of the natural history of the disease itself also. So if we are not sure about the progression of the disease and how it manifests itself over a lifetime horizon, there's another layer of uncertainty over there as well. So as far as that discussion has been in terms of what decision was, it was about cost effectiveness and the fact that the drug value in the longer term was not proven from the cost effectiveness modeling.
Yep. Sorry, Prem, want to make a comment?
I have a question for Dhruv. Great talk, by the way. So do you think future ATTR drugs are still likely to get a rare disease classification?
My understanding is that it's a function of the condition rather than the treatment itself. And so to the extent
but
that
we must be very close to the $200,000 threshold number.
I see where you're going with this. I think that in our first modeling analysis, we came to an estimate about 150, 170, so we were close already.
Yes.
And I suspect, you're right, I suspect that if we actually redid these numbers, it would be harder to prove that we would have fewer than 200,000 with the new wild type assumptions. This might be a question for our regulatory colleagues as to whether, if they had one drug in the class get through based on an orphan drug, can they reclassify the condition downstream?
Yeah, my understanding, maybe Norm, you can comment, is it's not what the estimates are about how many are actually diagnosed, and so we're not at anywhere near 200,000 in the U.S. diagnosed, but I may be wrong.
I think that's right.
Yeah. So at this point, according to Pfizer's records, I think there's only like 35,000 people taking tafamidis in the United States, so we're well short of the 200,000 that is needed. Dr. Masri, maybe you could just comment. You've written a lot about, the process of accessing drug for patients and what your experiences are. Is the process getting easier or more difficult? Is it hindering access? Are there particular populations that are at risk, like we were talking about earlier?
Yeah, I think we had kind of periods of time where initially it was difficult to get the drug, especially that about 30 to 40% of the patients would not get the drug, at least in our region, if it weren't for financial assistance, either in the form of foundation grants or free drug from the manufacturer. And then, in 2020, 2021, and 2022, this actually became really, really robust kind of an assistance. And then lately, things have changed a little bit and became, harder again. I don't think we've had any untreated patients before the beginning of the year, but this year so far I've seen 3 or 4 who cannot get the drug. And so I think there is now kind of a trend back to getting into issues. And it's a real issue and real problem. You don't want patient treatment to rely on the decision of having free grants or free drug or whatever decision is made. And this access is a real challenge. And honestly, unless you have a large program or you have the setup to support what Lynette and Megan talked about, it's very hard to do at the individual cardiologist level or neurologist or small program level. And finally, Dr. Kazi mentioned the changes that are coming in regards to the law, but I never know what these changes will end up doing, because I'll use the example of apixaban, where even though the out-of-pocket is not as high as tafamidis, yet most patients cannot afford it in our region as well, because of the rules and regulations regarding how much you have to spend before it kicks in. So all these changes end up kind of unpredictable, but patients rely largely here on financial assistance. And the hope is if we get more options to patients, that prices drop, but this is historically has not been the case.
There's one other change that I didn't highlight. The Inflation Reduction Act has a clause that if a patient has an anticipation of hitting $2,000 a year, so we think based on their drug use, they will hit $2,000 a year, they can spread the $2,000 equally through the year. So it'll be about $180 a month. Now, that's really important because the way Part D is currently structured is you show up in January with your prescriptions and you have your deductible sitting there in front of you, and then suddenly you have to pay your enormous deductible, so, which is one of the things that gets in the way of our patients on DOACs, because you hit the deductible just as soon as you start the DOAC. And so that's the other piece which may help some of our patients and help buffer the cost of the $2,000 through the year. There was an analysis that we did where we looked at what is a predictor of using more than $2,000, and this goes back to our equity conversations. Being from a minority racial ethnic subgroup is a predictor of not hitting your $2,000 cap. And that's not because there's less disease. It's because people can't even afford the $2,000. They're not even getting to that spend of $2,000. And so, again, I mentioned earlier that I worry about the unintended consequences. It solves some of our problems. It's a big step forward, but let's not pretend that it's going to fix the real equity issues in access to amyloidosis therapies.
Yeah, I echo that sentiment. I mean, and interestingly, in our data at Columbia, we have, 800 people on drug, and the time to actually get the drug delivered to the patient is 3 times as long if you live in a zip code that is, heavily populated with underserved minorities, even though none of those patients ever end up paying for the drug, right? So there's a discourse here between, or a discrepancy between the cost of the drug for what the patient's paying and how quickly they get access. Actually, the people who can pay for it, they get access very quickly. And I think part of the problem is that the process of trying to get access to the drug is so complicated, right? You have to fill out this form. You have to send in your tax returns. I mean, there are all these hurdles, and someone with not high health literacy really has a hard time accessing. Martin, you mentioned we could be prepared for the future, which is we're all hopeful that many of these newer therapies will be very effective as well, and so we could be in this enviable position of being able, we hope, as providers, to provide patients with the best therapy, that is maybe combinations of drugs. How do we, besides designing really good trials to give us the best evidence, is there anything you would suggest in that regard that we do now?
Yeah, there's lots of discussions going on at the moment, as you know, Matt. And I think in a way, it's like, what do we think treatment would look like in 5 or 7 years' time? And to your point, it's probably going to be depletion if that throws out silencing and stabilization in some form. But we have to construct the trials that give the evidence base for regulators and payers that whatever sequence we're proposing, we've got the evidence to support it. So I think we as a community need to think really carefully about 5-10 years hence and work backwards. And I think also that comes to endpoints. Everybody wants to do smaller trials, less patients, shorter, but there is a downside and a risk to that. So we have to get the balance right there as well. And I like the discussion also about, do we need placebo? Can we use historical? Can we do propensity matching? Can we use more clever AI to kind of synthetic cohorts? All of that is really mission critical to these discussions. So let's get really engaged and down and dirty with all of this and try and design the trials that will provide the evidence that reimbursement can't refute and the regulators are happy for risk benefit and safety. So it's not a direct answer, Matt, but that is exactly where we need to go.
Yeah, I would echo those sentiments. I think, further collaboration amongst centers. We talked about real-world data all day today. Maybe these are not going to be head-to-head trials, but we'll learn a lot from what patients are getting. I'm also reminded by the fact that not every drug stays on patent for forever, so these will become generic, right? And then, God willing, price will come down and access will, I think, only improve, so we'll be better able to help our patients. I thank everyone for participating, for fantastic presentations. I thank everyone for coming out, and I'll turn it over to Isabel for some final words.
Oh, yes, thank you all. And very briefly, we started today with Prem talking about the breathtaking developments that we've seen in the landscape and how today we were going to address some of the fundamental gaps, which I think we did well. A patient said it's an excellent opportunity for a better tomorrow. And it's really clear from everybody's participation today that there's a commitment and interest in advancing the field. So it doesn't stop here. The Amyloidosis Forum is committed to publishing, and we'll be publishing from the findings of the meeting today. We also, the steering committee, will reconvene and will develop work streams and working groups to really dig in on certain areas and priorities. Before I hand over to Dr. Adigan for the very last word, I would like to say that there is so much that goes on to planning a meeting like today and making it successful. And I really want to thank the steering committee who have gone over and above in giving their time, as well as the ARC team. And the person really who is the magician and the brains behind the curtain is Kristen Sue. And this really is, without her, this meeting would not only would it not be what it is, it would not have happened. So thank you. And so, Dr. Adigan, would you like to close the meeting?
Sure. Thank you, Isabel. I want to just echo your sentiments. Thanks to everyone, our virtual attendees, as well as everyone who's been here in person. I also want to specifically say a big thank you to regulatory agencies who've been here from the EMA, PDMA, and I'm sure they're no longer online, hopefully not. A big thank you to also my division director, Dr. Norman Stockbridge, for his support, being available for all the calls and guidance with this program. To all the panelists for being present and available for all the calls, the emails, the back and forth, we really appreciate you. And for everyone who's been here, I want to say a big thank you. In the coming weeks, I know ARC does a really good job of sending out some communication to get some work streams out and running. I want to encourage everyone. When we have meetings like this, everyone is excited, and we're looking forward to effecting change in the community. But what can easily happen is we go back and we're siloed in our different communities and organizations and institutions, that then it becomes almost like an unreachable target. But I think we've had some productive discussions today, so when ARC sends out emails and tries to engage us, I just want to encourage everyone to just be involved in the process, and let's come back next year and have some landmarks and things that we've done so that we can be encouraged and move this area of ATTR amyloidosis forward. And with that, I want to say thank you. And good luck with the traffic, with flights, and I wish everyone safe journey home. Thank you very much.